tuberculosis diagnostic tools. With PhD in medical microbiology 2011 from University of Delhi and an MBA in hospital administration, he is equipped with 8 years experience in TB laboratory monitoring and supervision on EQA, and 5 years experience in monitor and evaluate performance of laboratory network through all new electronic tools. He has been mentor and trainer for the implementation of quality management systems for 5 years. He has 3 years of experience about laboratory information management system besides customizing technical assistance as per TB laboratory. He achieved international auditor certification for ISO 15189 NABL, Delhi, India. His international awards for presentations in conferences in US, Vietnam, South Africa, Hong Kong are numerous and notable. Among them are Young Scientist Training Award on MTB, Mycobacterial Tuberculosis, Rifampicin, DTS, transfer technology to develop external quality assurance panel for gene expert from International Laboratory Branch Center for Disease Control Atlanta, that is CTC Atlanta, USA, and Young Scientist Award for oral presentation on successful implementation of DTS expert MTB Rifampicin EQA in Vietnam, in ASLM Cape Town, both in 2016. He is recognized as a best QMS expert to NTP from Ministry of Health, Vietnam in 2017. So I'd like to welcome Dr. Vidyanidhi Kumar to speak. Over to Dr. Vidyanidhi Kumar.
Yes, good afternoon, everyone, and very much thankful to Dr. Ramani Dhanraj, ma'am, as well as Dr. Veera Brahman, to give this opportunity to speak about the emerging diseases. So you tell me how much time is left with me, and according to that, I'll move the presentation.
You will have easily 45 to 50 minutes.
Thanks.
And then, or 45 minutes if you finish, then another 5-10 minutes we will.
Okay, so thank you, madam. So let me share my screen.
Yes.
Okay.
Thank you.
Yeah. So I'll put my, video is off because, the network may be down because of these videos. Can I stop my video? Is it okay? Hello?
Yeah, okay, sir.
Okay.
Yeah, it's good, sir. It's good.
Yeah. So thank you for my great introduction about myself. Okay, dear all, myself is Dr. Vidyanidhi. Now, today's talk is new diagnostic approaches, especially for molecular biology, in the field of molecular biology for diagnosis of TB and DRTB. So since most of the audience are belonging to the community of the biotechnology, so I think this is a relevant platform to speak about the molecular than any other technologies. So as you all know that now everybody is thinking about discussing about COVID. Morning 6 to the next day, morning 6 o'clock, you used to get all the WhatsApp messages, news and everything about the COVID. But you see, COVID is just an infant stage. It's just 3 years, 4 to 6 months old of disease. But in the case of tuberculosis, we have more than 100 years of experience and pathogenicity, and the most of the populations are severely affected with this tuberculosis. That's why when Dr. Veera Brahman is called me, then I said that it's okay. Now the COVID is, there are 2 more speakers are available. Let us speak about something about tuberculosis, which is very, very important to discuss and to know the things in future also. So you know that in 1882, the Robert Koch was invented, the bacteria of Mycobacterium tuberculosis. In one conference, he presented the paper. Then the entire world was looking at him. What was it? and why this disease is so important to discuss today as of today. Now, in my content of the presentation is having the brief background of the tuberculosis and what are the targets which was proposed by the WHO as well as the country management, and now what kind of laboratories are available across country, across the world, and what kind of diagnostics, which is very, very important to be discussed today's topic, and the brief short summary, what is a way forward steps. Now, as I said, the tuberculosis is caused by the Mycobacterium tuberculosis. It is a non-motile, a rod-shaped bacteria, and it is having 2 to 4 micrometers of length and 0.2 to 0.5 micrometers in the width. And this is an absolute and obligate aerobe, so it requires an oxygen to grow. And you know that the TB remains the world's deadliest infectious killer. Tuberculosis is a communicable disease that is a major cause of ill health. And you know that this is one of the top 10 causes of the death worldwide and leading a cause of death from a single infectious agent. It is caused by, as I said, tuberculosis. And each day, over 4,000 people loses their lives to TB because of TB. Do you know, I'll tell you the statistics, how many people are dying in India a day. And you know, the 30,000 people fall ill with this preventable disease and curable disease in a single year. And the global estimation is 1.2 million deaths occurred in HIV-negative people in 2018. So 2019 data is yet to come, yet to be published by the WHO. So right now, I'm showing some statistics which is related to 2018. So there is not much difference between the 2018 and 2020 as of our statistics, unpublished statistics. So I think this is relevant to speak about this disease burden across the world as well as in India. It is very shocking news if you look at the figures. Geographically, the most TB cases were recorded, especially in Southeast Asia, is contributing 44%. Among 44% in Southeast Asia, India contribution is 27%. Now you can imagine how threatening it is. Now, if you look at this graph, between this matrix was published in 2019 WHO report. If you look at India is accountable for a TB as well as TB HIV and MDR TB. So India is a middle and hot continent country which is having the left arm is an MDR TB is also on high burden as well as TB HIV. I will explain to you what is an MDR TB in the next coming slides, but it is a drug resistance, multi-drug resistance TB. So India is a middle hanger, which is holding both MDR TB and high MDR TB cases as well as TB and HIV comorbid condition patients. Now, let's talk about Indian burden cases. If you look at these figures, in 2018, the reports, only that year, 2.7 million, it means approximately 27 lakh population was recorded in the TB in 1 year. And 199 persons per 1 lakh population, which is a very, very high, considerably in other part of the world. So this is one of the key, key emerging and key surprise as well as threatening figures for the entire world. So as well as the mortality rate is also very high with the HIV negative patients, that is 440,000 is a high mortality recorded in a year. So this is somehow a statistical figures which was very much important to know everyone about what are the impact of the TB burden across the nation. Now, so WHO has set some of the goals it was suggesting all across the 174 nations. In 174 nations, there are 27 nations which are very, very important as having high TB burden. They set and they make some certain goals. That goals by 2035, the among 30 countries, which is high burden tuberculosis countries, should meet this target. So they suggested several suggestions, ideas to each and every country to reach these goals. As well as in India, I'm telling you, the political commitment from the PMO office to the field level is very high. Now the TB portfolio is directly looking is under the Prime Ministers of India. Previously it is under the Family of Welfare, even though it is a Minister of Family and Welfare, but regarding TB portfolio, the Prime Minister, Sri Narendra Modi, is looking very, very statistically looking into each and every aspect, and every month they are conducting the review. Okay, now you tell us what you have achieved in this month. So now you can see the country leadership is more focused towards to attain these goals. Regarding whatever the WHO has provided and set these goals. You know, what is the meaning of SDG? SDG means Sustainable Development Goals. So this is somehow the 5 years below the downline. So the WHO has told us every country that you should reach a 90% of reduction of the number of deaths comparatively in 2015. And reduction of the TB incidence rate is 80%. Now, the TB affected families facing catastrophic cost. You know, what do you mean by this catastrophic cost? Even though the person who infected the TB, then still we have a social stigma. Then he won't be able to go for a work. The daily wages, the most of the people are, you know, infected as well as affected with this cost bearing is very high and still in India. So WHO has made these goals. By 2030, if every country should reach these goals. So how to reach these goals? What are the criteria? What are the vision? What are the implementation strategies are required to reach these goals? I will tell you in the next slide in this. So in this anti-TB strategy, the WHO has declared that there is go for with 3 pillars. Even in India too is having these 3 pillars of strategies. One is a government stewardship, accountability, which is already going on. The second one is building a strong coalition with civil societies and communities. So the civil societies comes under not only the society, it comes under the R&D. You have to invent some new drugs. You have to invent some newer technologies to diagnosis as soon as possible. So these are very, very important to reach the goals. Then intensified research and innovations. You see, this is very, very important. Nowadays, and every the manufacturing industries, the diagnostic kit industries, as well as, you know, that the CSR institutes, everyone behind to invent some of the new technologies, new drugs to contribute for ending the TB by 2035. Do you know one of the interesting things? Even though the WHO said in 2035, but India has set a goal, you should reach that SDG goals by 2025. The Prime Minister said, I don't know whatever you want. I will give you the money, you do it a research, you make a policies, you implement the things. I want the results fast. So like in that way, the country management is going on. Now, let's talk about diagnostic methods which are available. You know, the microscopy. The microscopy, it is more than 100 years of invention. In 1882, Robert Koch was identified as bacteria under monocular microscopy. From that year to till now, this is also one of the point identification diagnostic step in the peripheral levels. But I will tell you, there are different sensitivities and specificities. And what I showed you in this slide, the limit of detection for a bacteria, if you want to see, identify and detect under microscopy, you required more than 10,000 bacteria per ml, then only it will give you a correct result. So the sensitivity is in about around 51% on an average. The simplest way, I'm telling you what is the sensitivity and specificity, and most of you know that the sensitivity is identifying the true positive, and the specificity is identifying true negative. This is very, very important when you are looking for any development of newer diagnostic tests. So as far as the microscopy concerned, it's a 51 percentile, and the specificity is 62 percentile. Now comes to the point, now the molecular diagnostics. Now this is a very relevant topic, and why we are choosing this molecular technology this today. I will tell you in the next slides. There are several molecular diagnostics are available. There are some of the diagnostic tests which was approved by the WHO, and what are approving years, which was narrated here, clear cut in the slide. And now my request to all the audience, if you say that if my slides are visible, I want from the audience, everyone is looking at my slides or not. This is one of the technical requirement from your side. So are you able to hear my voice and my slides? Someone say yes. Okay, I think there is some messages there. I hope everyone is able to see my slides. Okay. Dr. Veerabrahman, can you unmute and say something?
Yes, sir. Everybody watching your slides clearly.
Okay,
so no
clear.
Okay.
Thanks. Yeah, I need this clarity.
Yeah. Yes, sir. Yes.
This is during technical snacks maybe happen in between.
Of course. Thank you, sir.
Yeah. So if you look at the invention era and approved by the WHO, it actually starts from this microscope and LJ media is before 2007, as I said to you. And 2007, there was one more liquid culture method. As you all know that the bacteria, Robert Koch's postulate, you need an evidence. You have to culture something to grow something, whether it is in vitro or in vivo, then it is a must for the proving a true positive and true negative. And from the 2008, the the development, most of the development was happened in the case of diagnostics for the tuberculosis is from the 2008. You can say that from 2008, it is a good time for the tuberculosis patient for getting a results in a very short time. I'll tell you the interesting one of the things. Once the patient reaches to a doctor and doctor go for, okay, he will see the clinical findings. Based on clinical findings, he will suggest for an X-ray, then go for a microscopy. Then the sensitivity and specificity is 51% and 68 percentile. Then based on those evidences, the doctor used to treat. So sometimes because of low sensitivity and specificity, doctor may go wrong and misinterpret as a tuberculosis patient. Maybe the patient without treatment, he was declared as it's not a TB. That is one of the bad things or maybe the limitations of the previous technologies. And once the patient comes to the doctor and go for a culture, that is a solid culture, it takes time to grow, and the report comes to after 56 weeks. So there is also another challenging is patient will not come. He will say that if I go to the government doctor, he will keep on lingering on me and he will give after 56 weeks a result. Then by that time, the management of the patient interest towards taking the drugs, everything will be lost. So that is the reason I said from 2007 or 2008 onwards, there is a golden days for the tuberculosis patient as well as the doctors. They can able to give the results as soon as possible. So these are some of the WHO approved diagnostic tests, and they are the chronological order of the approved years. If you look at the 2010 and 2020, then almost 10 years, it took time to approve newer technologies. It is a big time, but still there are certain limitations in the molecular biology as far as concerned for the TB test. I will tell you why these limitations. Now let us speak about India. There are several laboratories. How many people know that if the patient is tuberculosis, where to go, and what kind of test needs to be recommended for this patient? Even the most of the clinicians are required to know. So the central government, the Ministry of Health, has given a clear-cut suggestions, as well as given information to them across the country and trained the people. And if the patient comes to you, what kind of test to be done at which level? So there are several tests at community level. There are wellness centers. Now the people used to call it as the wellness centers. It is previously it is called as community healthcare centers. There, the government has established a rapid molecular test, and at the community level. So the patient can reach us immediately to the facility to get it done, the test, as soon as possible. And regarding this district level, there are some higher methodologies, molecular testings is implemented and they're installed at level. So now you are from Visakhapatnam, you have a very good and beautiful, very quality management system laboratory, which is near by you, that is opposite of Pedavalter, that is TB and chest disease. That is an intermediate at district level laboratory, which is having all capacity of molecular as well as the routine TB diagnostics. Then there is another reference laboratories, which is required for a typical cases. Even at the intermediate laboratory, if some of the drug resistance are unable to test at intermediate reference laboratory, they will send for the reference laboratory where some of the extended drug susceptibility testing will be done by a different molecular test, including a genome sequencing. That is a very important step towards ending the TB goals. So these are some of the laboratory network in India. So now let us speak about the molecular test. Okay, now you see the choice of the test is based on the rapid result and more sensitive and specific. Now this molecular biology, the rapid diagnostics, as I told you in the previous, the TB program is mostly starts in 1997. And initially they used to get the report, the patient used to get the report around 56 weeks. Now the 56 weeks comes down to a minute. If you look at the credentiality and credibility of the molecular biology. Now there are several molecular testing platforms which are available, and there are different drug targets. There are different gene targets, which was having a high sensitive and specific, and those targets who are recommended for doing this molecular test. So there are some of the IS elements, insertion sequence 6110 insertion element is there, which is mostly available in the tuberculosis. There is also another target, 16S rRNA, HSP65, and there are 23 sRNA. So all these things are available in the reference standards. You can see in WHO website, they will give you, there is a big, beautiful document, what kind of targets are which are more sensitive and specific for diagnosing of tuberculosis. So this is one of the important and good golden things, the usefulness of this molecular biology, uses of the molecular biology for the TB. Now, let us speak about a genome. What is the length of the genome of the MTB? This is a 4.4 million base pairs of the long of Mycobacterium tuberculosis, and approximately 400 genes. They are the encoding regions which are very active and which are involved for the tuberculosis. When the bacteria wants to replicate, they require the 400 active genes which are there, which are so essential for the growth of the Mycobacterium tuberculosis in intracellular. But the 4.4 million base pairs are there. When there was a whole genome sequence, I will tell you what is that whole genome sequence in the coming slides. Now, one by one, what are these technologies? Okay, I told you, I have given a table, okay, these are the technologies. But what is the principle behind this? This is very, very important. Now, this is first as a sequential order. ICAP is a gene expert is one of the important findings, one important innovation by the US. I will tell you shortly of this gene expert story. If you remember, there was an era of the bioterrorism. There is some of this Bacillus anthrax. They used to send the US in the powder form by different other nations. Then the government of US, come into the things, okay, let us invent some new technology to identify this powder format, which can be, the technology can be placed in the postal department so that, whatever the powder which was received in the parcel can be tested whether it is a biological agent is there or not. So that is the first initiating step for a gene expert for invention. Later, this gene expert technology was, enlarged, implemented, and scattered for a various diagnosis. It's more than 47 different varieties of diagnosis it is using right now. So one of the diagnostics infectious agent is tuberculosis. Now, if you look at the gene expert mission, this is an assay target is an RPOB gene, as well as for the detection of the rifampicin resistance, as well as the target gene is IS6110. There is another target which is for the diagnosis for the tuberculosis. There is also an updated version came, Gene Expert Ultra, which is having a target. They've added one more target that is 1082. I'll tell you what is that is 16S rRNA target, which is also be covered when they are doing a PCR reaction, so that the sensitivity and specificity will be more. The true diagnosis, the true testing can be happen for a right sample or the right patient. Now, this cartridge, the Gene Expert cartridge, is having a different components. One is the syringe drive, a cartridge body is there, PCR reaction tube, PCR reaction tubes. There are several, there are several parts is embedded in this cartridge. So it is also available in the internet. You can see later of this presentation whenever you have a free time. But this is a very unique model of this cartridge. Once the patient sample received by the lab, what the lab needs to do is simple. They need to add this sample reagent to a particular sample. Now, someone can ask me the question, what kind of samples are acceptable for doing this test? You see, if you are looking for any diagnostic test, the right sample is very important. The right sample at the right time, if you do, then the sensitivity and specificity of the diagnostic test will always improve. So this Gene Expert testing, the sputum, the tuberculosis patient, the pulmonary tuberculosis patient, the sputum is an appropriate sample. Apart from the sputum, you can also test in other samples like gastric lavage, the cerebrospinal fluid, or the pleural fluid. There are several recommendations were given by the WHO. Those samples can be used for the diagnosis of tuberculosis. But the more sensitivity and specificity is on sputum sample. So the pulmonary patient, which is having an expectorant sputum, then he sure chance that he can able to get a right result. So it is a very simple technology. Once you receive a sputum sample from the patient, you just add the sample reagent. Then after 15 minutes, there is a processing methodology. Just take 2 ml of in that and add it into this cartridge and put into the machine. That's it. It is, while you're doing a test, if the minimizing the steps, it will also minimizing a risk of cross-contamination. So this is a very simple step. It is a 2-step process. One is the adding of sample reagent. Once adding of the time sample reagent, after 15 minutes, take 2 ml of the sample and add it into the cartridge so that you can minimize the cross-contamination. So I'm loaded into the cartridge, loaded into the machine. So right now I'm going to show you a simple video which what kind of reactions will happen inside that cartridge. So I'm going to start a small video on this. So this is the GeneXpert cartridge, and it has, there are 3 different. One is a wall body, reaction tube, and the top. And it has a beaded, which was there inside in the cartridge. Those cartridges are ready for use. Once you add a sample into that cartridge and put inside the machine, so now the sample will go for further process. There was a plunger, it will aspirate that sample inside, then again rotates, remove all the debris and send a bacteria to a lower portion of the filter. So all the bacteria are interacting with this filter. The filter has an affinity so that the bacteria is stick to that filter. The later, there was an ultrasonication method which the bacteria will be lysed and the genetic material will come. If the sample contains the tuberculosis bacteria, so the genome will come out from that lotion. Then again, the DNA will be pushed into another chamber, which is having a reaction with primers, and it will push to a reaction chamber. And once the PCR will be done with several cycles, then once the PCR reaction was happened, then the fluorescence emission will be occurred. Once the fluorescence emission will be occurred, then that will be detected by the machine and will say, yes, it was positive. If the fluorescent emission was not happened up to certain cycle threshold, then it will show declared as a negative. So this is one of the classical video pictographical representation, how it seems to be. Once it has happened, then it will be come out. So this is how the work progress inside the machine. Now, okay, I told you that how the process will be happen inside the machine, but what is an actual principle behind this? If you look at, as I told you that there are certain targets, which I said that IS 6100 as well as the RPO gene, these are the targeted and they will be amplified. There are 2 molecular beacons will be there. They are the fluorophores that will quench and added to the primers. Once the cycling condition, once the PCR will be, once the PCR cycle will go on, then it will be detected the fluorescent emission. That fluorescent emission will be detected by the machine. So this is the principle behind the GeneXpert. If there is no target, if the PCR reaction hasn't happened, if there is no targets are available, it means that there is no Mycobacterium tuberculosis genetic material was not extracted by the system. It means there is no fluorescence emission. If there is no fluorescence emission, it means there is no detection of the tuberculosis. What are the advantages? The advantages is you can see that the time to result is below 2 hours, and you can test 4 samples in one go, 4 different patients. There are different varieties of the gene expert machines are there. You can do for the 2 patient, that is called as 2 module, 4 patient, 4 module, 16 module, as well as 64 module machines is also available. So based on the workload, based on the patients, so Government of India right now 16 module machines were installed in way across the country. So it means at a time the 16 patients will go. So in a day you can do more patients. The specificity and sensitivity is a 98 percentile. 99 percentile is a specific. As I told you, the sensitivity is 98 in the smear positive, 72 percentile in the smear negative cases. So that is an advantage. Now, as I told you that there was a 10 years gap was there in invention between 2010 by the gene expert. There was another methodology which was endorsed by the WHO, that is a Truenight. This is also a based and nested PCR, okay, and that was endorsed by the WHO. And one more thing is, this is developed by India. This is one of a key point to say to everyone. Now the India is giving a lot of efforts, putting a lot of efforts to developing so many rapid kits. So this is called as a Truenight. You can detect tuberculosis, as well as you can also detect rifampicin. So this is a very simple method. It is also again a 2-step. There is a 2-step process. One is, if the patient comes to a doctor, will give you a sputum, then first they will identify whether it is a sputum, whether the tuberculosis is there or not. Once the first step, the tuberculosis is identified, there was another step is there to detect whether the patient is drug resistant, particularly towards the rifampicin or not. So this is somehow a 2-step strategy, but in the case of gene expert, it's only 1 step. Just put it into the cartridge, keep it inside, and then after, in around 2 hours, the result will be whether the patient is TB, as well as whether the patient is sensitive to a drug of rifampicin or resistant to a drug of rifampicin. So there are small differences between the 2 diagnostic kits, but it is also very good sensitive and specificity. So now there is one more interesting point is now in Visakhapatnam, the Truenight. In Visakhapatnam, Truenight testing kits are, what I came to know that they are now manufacturing at Visakhapatnam. But maybe for the COVID, so in future maybe it will for tuberculosis. So it is more and more easy to get the kits in time. So simple test, just you require a 4.5 microliter to add it into the chip base. This is a microchip based. Once the microchip, it will go inside, it will do all its DNA extraction will be done, the identification of rifampicin resistance, and it will give you a result. So all these things will happen with this Truenight. Now, what are the advantages? The advantage is, as I told you, that the TB testing is results available within 60 minutes, less expensive, and as well as the sensitivity is also equivalent to 89 percentile for the MTBSA, and specificity is 98. So for the rifampicin detection, the sensitivity is 93 and specificity is 98%. Now, these 2 are very good technologies and will give you a result as soon as possible. And these 2 systems can be placed at community level so that you can have a less expensive, a less infrastructure, so that the people can reach to the peripheral level to get the things done as soon as possible. Now, as I showed you this pyramid, there was an intermediate reference laboratory. In the intermediate reference laboratory, there was another molecular test which was also be implemented by the Government of India, that is line probe assay. This is a line probe assay. It's very interesting. Actually, this was the first pioneering for the molecular development for the tuberculosis. But it was developed in 2008, and it was endorsed by the WHO. Then it has somehow different steps. There are multiple steps are available. You have to do a DNA extraction. It's a manual process. Again, you have to do amplification by the PCR. I will tell you what kind of steps are there. Then hybridization. This is a membrane strip technology. There's a hybridization. You need to do that. And the fourth step is an interpretation result. So what are the main advantages of this? Someone can say that, sir, you have a very simple technology that is GeneXpert as well as the Truenight, and the results are available within 2 hours. Then what is the requirement of implementing this technology in the intermediate reference laboratory? I will tell you a very, very important, this technology. So in the previous 2 technologies, you can identify only one drug, which is rifampicin. Even though the rifampicin is a key drug requirement, the sensitivity profile for initiating the treatment for the patient, but apart from that, now there is a new treatment guidelines came into the scenario. So the doctor requires other drug profiles like isoniazid, which is very important. Again, the fluoroquinolones, that is a moxifloxacin, aminoglycosides, capomycin, amikacin. These drugs are very, very important for the doctor to initiate the treatment, whether those drug sensitivity profiles are required. So which is not possible with the previous technologies as of today. But what I heard that in next couple of, maybe in by the 6 months down the line, those drugs is also can be identified with the previous technologies. But as of today, it is not possible with those 2 technologies. With this technology only, you can identify those drugs. Now, it was, as I told you, that there is a multi-steps are involved. One is, there are manual steps. One is you need to go for a lysis and you have to extract the DNA. This is one of the first steps. Second one is you have to put the PCR. And the third step is doing the hybridization. What is this hybridization? This hybridization is the DNA probes were there in the strip. Once the DNA was identified, extracted, amplified by the PCR, then it will, hybridized on the strips. Then it will give you the color bands. Look at, this is the step 4. The color bands will be coming like this. So there is an interpretation methodology is there. Based on this probes, you can say that whether the patient is a TB as well as the resistance to various drugs. What I told you, this with this help of this LPA, you can identify the sensitivity profile, whether it's a sensitive or sensitive or resistant with the rifampicin, isoniazid, fluoroquinolones, as well as aminoglycosides. In aminoglycoside, there is a category of, that is a 3 more drugs, that is amikacin, capomycin, and kanamycin. So all these drugs can be identified through this technology. That is the reason this technology was established in the intermediate reference laboratory. So the challenges with these technologies are, you required a what you can say, you required a very good infrastructure. You would require biosafety level 3 laboratories are required. So the maintenance, the cost expensive, and all these things are involved. But yes, in our scenario, patient is the most important. Patient, we can afford this test. Nowadays, the test cost is in around 1,000 rupees per patient, so which is affordable for government of India. So now all these tests are free of cost. Let me tell you, the government of India become free of cost for the patient. All these tests can be done free of cost. These are the advantages. It's a fast, less biohazardous risk. You can go with multiple drug targets. What are the disadvantages? As I told you, that the disadvantage with this step is you have to be very careful when you are doing a number of samples. There is a risk of cross-contamination. If you have not written a proper patient ID on the vials, then there is a chances of the miss or wrong or transcriptional errors during this implementing, doing testing with this technology. So sometimes there's a cross-contamination, a wrong result goes to a patient. So this is very, very important when the person is doing in this technology. He has to be very careful, do the less mistakes. So there is one more upcoming technology is coming. It is not yet to be endorsed by the WHO, but still it is a fluorescent-based fluorotype technology. It is also on an upcoming diagnostic test. It is also a molecular-based test technology. It will also emission the fluorescent light. Based on the fluorescent light, it will detect whether it is MTB positive or not. And still, it is also a limitation. It can be done for only 2 drugs. One is for INH and rifampicin. Still, this method is valid under the validation. Now, all are biotechnology. Most of the audience are belong to a biotechnology community. Now, everyone is looking at this whole genome sequence, targeted sequence. Now, the government of India installed 6 sequencers at reference laboratories, where the target sequence as well as whole genome sequence is possible. So some of the patients, there are very, very important, some of the patient, doctor is also unable to identify, yeah, everything is sensitive, still the patient is not responding with the drugs. Okay, everything is, then there is another scenario, yes, okay, patient is resistant to so many drugs, but I don't know what kind of drug needs to be modified. So they will send for this either targeted sequence or whole genome sequence so that the reference laboratory can give the suggestion for what kind of drugs can be suggested, what kind of drugs can be given to a patient. So that is the goodness of this whole genome sequence. Now the whole genome sequence was placed at 6 places. Now already few labs already started giving reports based on the sequence. So the next generation sequence, this is a Illumina MiSeq, the system. In this next generation sequence, you can do for whole genome with this machine. This is also has the different steps. One is you require a sample extraction, then you have to prepare a library preparation, which is very important step. Then you load into the sequencing machine. Then the data analysis will be. So my suggestion for the community, the audience, if the person who is studying the biotechnology, please concentrate future for bioinformatics. Now the next download the 10 years, the next year, this thing, bioinformatics will play a crucial role for the laboratory aspect, as well as the clinical management aspect, as well as core research aspect. So these are one of my suggestion. What I said, the bioinformatics is very, very important, and it's a very good scope of for the job perspective also. So, as of the last before one of the slide, knowing is not enough. We must apply. Willing is not enough. We must do. This is very, very important. And my last suggestion, the last summary of the slide is, still there is a lot of scope to invent for the newer diagnostic technologies. The way 1882 to 2020, the approach from 3 months diagnostic result to 2 hours to less than 1 hour diagnostic result. Still there is a scope. You can invent a newer technology maybe in 15 minutes or 20 minutes. So this is a very important and it's a very good scope in the molecular diagnostics. Thank you so much.
Very, very good. Dr. Veerabrahmam has questions down the line, Dr. Vidyanandi. Yes.
Yes.
Yeah, actually, I'm unable to see the questions.
You can see in the question Q&A box, sir.
Where is some question?
Yes, and
okay, sure.
Yes.
Can you please tell me the test other than PCR based to diagnose TB? Yes. Perfect. So, okay, let me go through the one of the slide which I showed you before. So you look at this. there was different technologies are available. One is the smear microscopy, that is one of the diagnostic tests. The second one is the solid culture as well as the liquid culture. So once you receive the sputum sample, you have to go for a decontamination, some processing methodology. Then you just inoculate it into the culture. And if the TB is positive, if the patient sample is having the tuberculosis bacilli, it will grow in this particular medium and it will give you the result. So as of today, the rapid culture method, that is called as a liquid method, that is MGIT, that is microbacterial growth indicator tube. You see, this is a liquid culture. This is the methodology which is there. Are you able to see my screen? Hello?
We're not able to see it.
Oh, sorry.
I think because you must have disabled screen sharing.
Then yeah, I could have told me that. Okay. There was another methodology apart from the molecular biology. Since today's topic is mostly on the molecular biology, that's why I was so concentrated on that. But there is other methodologies. That is a smear microscopy is there. The culture methodology is there. That is LPA, liquid culture. That is MGIT, microbacterial growth indicator tube. That is a mission. Once you inoculate it into that culture, then you have to keep it inside that machine. Once the culture grown, it will give you a beep. Then you have to identify it with further with the rapid antigen kits. So there is other methodologies also be there apart from the molecular biology. There are several PCR-based, there is in-house PCR-based methodologies are there, but whatever I'm telling you, that is a WHO-approved diagnostics, which is having some authentication as well as having the scientific evidences of their sensitivity. Yes, any other questions?
Yes, sir. Yes, the other question is there in chat box. So what are the molecular tests for the diagnosis of TB on latent phase?
Okay, right now the latent TB, LTBI, there is right now there is serological tests are available. A molecular test for the latent TB is challenging right now because we all are BCG vaccinated, as well as India is in a high TB burden country. So most of the people are having some latent TB infection. So the sensitivity is a big issue with the molecular technology, as well as in the latent TB, there is one of the disadvantages with the molecular technology is even though the bacteria is dead, if the patient is an asymptomatic or bacteria is dead, still it gives you a positive. So regarding these challenges for the latent phase of the TB, there are some technologies are under review, under validating by the WHO, but still there is a serological method that is there is ELISpot, a 24-hour technology is there. Even the government of India is also willing to implant this technology in the intermediate reference laboratory, but still the decision, still these discussions are going on. There are some sensitivity issues are there. So, but as of my knowledge, the molecular test for an LTBI is not yet approved by the government, the WHO, or for any program, any other country programs. Yes.
Okay, wonderful, sir. Perfect.
There are not too many questions, so there are 2 hands raised. Shall I allow them, Dr. Vidyanidhi?
Yeah, sure, no problem.
I will allow Dr. K. Sunita to ask her question. Dr. K. Sunita, please unmute your audio and shoot your question to Dr. Vidyanidhi. And then I will allow Dr. O. Sivapriya, last 2 questions, then I'll ask. Then maybe at the end we can ask Virubrahman. Is Dr. Sunita on the way? Dr. Sivapriya has unmuted herself. Let her ask. We will allow Dr. Sivapriya to speak. Dr. Sivapriya, please unmute your audio and ask the question. Dr. Sivapriya, are you there? Please unmute and ask. Okay, by the time they unmute, Dr. Vidyanidhi, I'm a social scientist. Yeah, Dr. Sunita is there. Let her ask the question. Dr. Sunita, please ask your question.
Sir, your session is very important and it is giving lot of information. Thank you, sir.
Welcome.
Okay.
So you have any question?
She's not.
No questions, sir. No questions. Already they asked. Previously you told it.
So when your session is so clear, I think Dr. Vidyanidhi, there'll be you're so clear. My question is, as a social scientist, of course, I don't know so much of biotechnology like you and Veerabrahman, but I've read somewhere that this Truenight test is really good, but it basically works on a DNA model, this molecule,
Yes.
no? So, but
Yes.
whereas the SARS-CoV-2, which causes the virus, is an RNA. Now, how
Yes.
quick is the RT reverse transcription PCR process, and is it so effective?
Ma'am, as of today, there are several good reports. Even ICMR has also approved this technology. Okay, they have a very, even the real-time PCR, this is, sorry, reverse transcriptase PCR, they have, there are two things. One is for the screening and the another one is for the confirmation. So very good sensitivity and specificity in that regard. I think there is a, I think Dr. Jogeshwar has that slide regarding this Truenight. There are several targets which were, yeah, there are several targets. Is there, remember, if Dr. Jogeshwar, please correct me if I'm wrong, there is an N antigen and E antigens are there. I think they are developing those targets.
Maybe he'll touch on them. Yes.
I think he will definitely, yes. But real time, yes, Truenight is very good technology. But we should appreciate.
I know,
yeah.
I've been reading that even remote parts of AP and all, Gadichalli and all, they're sending because it's cost only 1,300. But do you think with the number of COVID cases increasing, we'll be able to match the number of required supply demand? What do you say?
Ma'am, that's why I think Truenight has installed their company in Vizag to increase the manufacturing, increasing this manufacturing. Yes, I do agree the supply chain, the demand and supply, it still is a gap.
Yes.
I hope Truenight is mitigating those plans.
So good. So very happy, Dr. Vidyanidhi, and very interesting, though I caught every word of what you said, except when I went out just for a few minutes to have lunch. But it is, as you said yesterday, you promised, the way forward was very clear in your session, and we're very happy. On behalf of the principal, Dr. Sjcw, and the college, we offer our thanks and namaskar.
So my suggestion to your biotechnology department, if possible, initiate regarding the bioinformatics some programs, which is very important.
They have done, I think, they have done even a workshop in conjunction with IIT Bombay, and they have done. So they'll take it forward, as you said.
Yeah, that is very important. Now the future is with the bioinformatics.
Yes, yes. So very big namaskar to you. Thank you so much. You're a young researcher and scientist. We're very happy. We heard a lot of young, fresh ideas. Thank you very much.
Thanks, sir. So if you don't mind, can I log out, because I have another session. I have another operational review call with Geneva.
Yes, yes, please.
Thanks. Thanks to everyone. Thanks, Veerabrahman and Dr. Jogeshwar. Thanks.
Yes, sir. Yes. So very wonderful talk and informative talk by the Dr. Vidyanidhi Gumma. On behalf of St. Joseph College, we'd like to thank you for your wonderful talk. Thank you, sir.
Thanks. Thanks.
Now I'd request Dr. Veerabrahman to introduce our second speaker, who's already ready. He's listening probably, Dr. Jogeshwar Gabi. Dr. Veerabrahman, please introduce him.
Thank you, ma'am.
Thank you.
Dear participants and resource persons, we are proceeding to session 2. I've been introducing Dr. Jogeshwar Gadi. It is a pleasure to introduce Dr. Jogeshwar Gadi, Director and Scientist, Sjcw Molecular Diagnostics and Research Center, Sjcw. Has a PhD in genetics from Osmania University, Hyderabad. He worked as a research scientist and associate professor in AU College of Pharmaceutical Sciences, Visakhapatnam from 2013 to 16. He took up teaching Associateship followed by Assistant Research Professor at the Institute of Endocrinology, Ansei University, College of Medicine, South Korea for 6 years. Dr. Yogeshwar worked as APNL Project Fellow, APNL SRF, and CSIR Senior Research Fellow at beginning. He's received many awards from many organizations, including travel grants from Korean Endocrine Society, Best Oral Presentation from KS, that's Korean Endocrine Society, CSIR SRA, Pool Scientist Fellowship 2013. Young Scientist Referee Award, Biomaterials from Journal of Biomaterials, Elsevier, 2008, to name just a few. Dr. Gari is a brilliant researcher, as is evident from his CV. He is a reviewer of many well-noted journals like Journals of Biomaterials, Journal Pharmaceutical Biology, International Journal of Pharmaceutics, Journal of Biomedical Materials Research since 2008, 2010, and 2011, respectively. Besides being editorial board member for the journal Endocrinology and Metabolic Syndrome currently. He is a member of many scientific societies like for Korean Endocrine Society, American Society of Bone and Mineral Research, International Society of Endocrinology, Korean Anatomical Society, to name just few. We feel privileged to have Dr. Yogeshwar as a resource speaker for the one-day webinar in biotechnology in St. Joseph College for Women, Autonomous, Sjcw. I now invite you, sir, to talk over the session. Over to Yogeshwar Gadi, Dr. Yogeshwar Gadi.
Hello. Can you hear me, Dr. Veerbrahman?
Yes, sir. Yes, sir. I'm here.
Yeah. Please start video. Yeah. Please start video.
Yes, sir.
Yeah.
You may, yeah. We've given you co-host, you can start.
And now you can see, sir.
Yes.
Yeah.
Yeah, it is visible, sir.
Yes.
Yeah. I would
You
like
can start
to
sharing your screen also.
the main.
Sir.
Hello.
Are you sharing your screen? You may start that, so you'll be seen and your screen will be seen also.
Yeah. I already started.
Just check, sir.
Is it fine? Can you see me?
No, we can see you, but your screen is not visible. Sir.
Okay. Can you see now?
We can't see your screen, sir.
Okay, wait. Now?
Yes, it's started. So we'll be able to see your screen.
Yeah.
Yes, sir.
So I would.
You put it double click, you'll go into slideshow mode and
Yeah. So I would like to thank Dr. Veerbrahman.
Thank you.
Hello. Is it fine? Yeah.
Yes, it's fine, sir. Please go on. Thank you.
Yeah. I would like to thank Dr. Veerbrahman and other faculty members of St. Joseph's College for Women, Jnanapuram, Visakhapatnam. I have a strong association with your college that I am sometimes coming as a board of studies member for biotechnology. And it's a privilege to serve in such position in St. Joseph's College for Women. And today, actually, I would like to have some general conversation about this meeting because actually last 2 months, I'm tired in working at King George Hospital, Visakhapatnam in COVID-19 project only. So actually when contacted me, Dr. Veerbrahman, he said, actually, sir, we need to conduct this meeting. So please guide me. So when the time we look about the present scenario of what these are all diseases we are seeing this emerging novel diseases, and we have a lecture from Dr. Vidyanidhi about TB and its resistance. Now coronavirus, and so we don't have solutions at this moment. Actually, these are all novel diseases. Even people don't know how to treat and how to control and all. So with this, so it's very interesting. I told Veer Brahmam, yeah, go ahead, so that we'll have the students and all the researchers will get benefited. At least some clues will come out from these kind of lectures, so that the people will think about the diagnostic patterns and also some kind of inventions about the novel drugs for treating these kind of viruses or bacteria like Mycobacterium tuberculosis. So like that we started, and finally it comes this day. And really, I'm very busy in hectic work at KGH. We are testing a day at least 1,000 samples of COVID suspected people, and also those who are in quarantines and those who are in community-based studies. And we are really very busy. Apart from that, I came here to speak. At least I would like to share my knowledge and my experience in last 2 months, and so that the biotechnology community and biotechnology students and also researchers get benefited. So title of my presentation is Molecular Diagnostics for Detection of Novel Coronavirus. Actually, this corona family, coronaviruses, these are all actually the SARS virus, and also Southeast, Middle East MERS, Middle East Respiratory Syndrome, and SARS, Severe Acute Respiratory Syndrome. These are all belongs to the same genus. These are beta virus family. And this is my present workplace, where I'm working as senior scientist, virology laboratory. It's a contractual position, and the government asked us to participate in this proposed project, and we joined in March 12th. We joined there in COVID-19 testing at King George Hospital, Visakhapatnam. And coming to the story of coronavirus, as you're all aware, I have seen in the newspapers every day we are thinking about coronavirus. We have coronavirus fever. So it started its journey in December 2019. The cluster of pneumonia cases. It's a non-unusual pneumonia in China, which is due to some unknown virus. They have present that time they just think this is an unknown virus, and they named it as a coronavirus 2019 because it originated in 2019. And its corona, why corona means its structure. Based on its structure, they named it as a coronavirus. It's the spike proteins on the structure looks like crown shape, so they have named it as a coronavirus. So this is the typical structure of coronavirus. You can see the spike glycoproteins and RNA and N protein and envelope protein and hemagglutinin esterase dimer. So this is how it looks like corona. So that's why they named it as coronavirus. It looks like crown shape, sorry. Okay. And coming to the origin, so we see here the different types of coronaviruses. Actually, the coronaviruses are, as I mentioned earlier, SARS, MERS, and nCoV. There are also other types of coronavirus. And the basic symptoms of coronavirus based on their response, like pathogenicity, like respiratory, they cause common cold, pneumonia. And coming to the gastrointestinal symptoms, such as generally it causes the mild disease. And some causes severe diseases are SARS, that is also reported in China, and MERS, it is Saudi. They have Middle East Respiratory Syndrome coronavirus. They called it as it's originated in Saudi Arabia. And nCoV-19, which is also originated in China. The variations among these, because these are all belongs to the same family, you can see genome's comparison of different, this is recent publication in 2020, some review article. I take this from some review article. Here you can see this is the 2019 nCoV, the structure. It has actually the size of the whole genome of coronavirus is about 26 kilobases, 26 kilobases to 30 kilobases. It's 31.6 kilobases, 26.6 to 31.6 kilobases, the size of the genome. And it has 6 proteins, 6 ORFs, sorry. It contains 6 ORFs and constitutes so many number of proteins. And you can see the comparative, this is a phylogenetic tree of these genus, entire genus of coronaviruses, and where we have only 3 kinds of coronavirus are very dangerous to the humans, such as 2000 nCoV, 19 nCoV, SARS-CoV, and MERS-CoV. These are all human harmful diseases they cause. And this is the 3 comparative, because there are only slight variation between COVID-19, SARS-CoV, and MERS-CoV. You can see the sizes. SARS COVID-19 is 29844 base pairs, 29.8 kilobases, and SARS-CoV is 29.751 base pairs. MERS-CoV is 30.119 base pairs. And the only in the variations at these E, M, and N proteins, envelopes or internal proteins, this variation causes the severity of the disease. You can see here these E, M, and N regions are very, very important regions which are important in the pathogenicity of these individual viruses. And this is the article. People who are interested, you can go through this genotype and phenotype of COVID-19 and the roles in pathogenesis. You can get more details about this. It is a review article. It's excellent, and very recent information is there. Those who are interested can go through this article. And coming to how corona transmits to the globe. Hope all are at home, and all people enjoying in the home, and because of this only we are having in web meeting. Otherwise we have direct, we might have in the direct meeting. So see how corona transmits. See, there are group of people, wherever the group of people. See, in China, first, see, in Wuhan, seafood and live animal market in Wuhan, the people who are suffering with severe pneumonic infection, pneumonia, severe pneumonia. So those who person causes, that is unusual pneumonia. It's a not usual pneumonia, it's unusual pneumonia. So who is sick and who transmits to his family members and healthcare workers. There the chain starts. It's a pandemic. Okay. So it started from China and traveled throughout the globe, and it contaminated 216 countries. So you can see many cases spreads in China and to other countries by air travel or other means. So that's why stay safe at home. And so we'll see what are the symptoms, diagnosis, and treatment. Anybody knows? Can anybody answer? What are the diagnosis, treatments, and symptoms? Hello, can you hear?
Hello, sir. Hello.
Hello.
Sir, it is audible. Continue, sir.
Yeah, is it going well? Okay. Which slide? It's symptoms, diagnosis, and treatment. Is it fine, right?
Sir, here there is no screen share, sir. Please share the screen.
Okay. Where it stopped?
It
Can you
stopped
tell me?
fourth.
Video also stopped, sir.
Yeah. I'm just
Just
starting.
share the screen, sir.
Yeah, video is fine. Okay.
Video is good, sir. You can share the screen, sir.
Yeah.
The symptom slides, sir.
Yeah. Okay.
Okay, sir.
Yes, fine,
Yeah.
sir, fine.
Yeah, the symptoms, diagnosis and treatment. So the symptoms are mild to severe. There are fever, cough, shortness of breath, pneumonia, kidney failure, and death. Mortality rate not determined yet. And diagnostics, the only way to do diagnose this COVID-19 is PCR. And there is also another test, that is actually ICMR has stopped doing that rapid test. It's still under validation. And the treatment is no specific medicine, supportive care, and vaccine. So people are really exciting to get these specific medicines for COVID-19 actually. And prevention, how to prevent COVID-19. So covering of mouth and nose, avoiding close contact with persons who are sick, appropriate use of masks and PPE, hand washing, stay at home if you are sick, avoid contact with animals, cook well when you eat different meat. And coming to the diagnostic part, this is actually now very rapidly doing across the country and also across the globe, because if we need to identify the people who are really having, because nowadays you might heard in the newspapers that there is asymptomatic corona rather than symptomatic corona. Why such things happen? As you are seeing the genome of coronavirus, it's an RNA virus. So it is varying, the rapidly it is getting mutated at the RNA level. It is getting mutated and it is varying its structure also. The structure is the continuous, the round spear structure is very important for the pathogenicity. That means if it is, luckily we have in our state, actually in India, some province having the very dangerous variant, that is A2A variant. Some people say it's a A2A variant. And in our Andhra Pradesh, we don't have very dangerous variant. That why I'm saying not dangerous means, it's don't think that you should go into the people and you should contaminate the people even if you have asymptomatic virus. It's not that, because it is varying in person to person. That depends on the host to host. It is varying its structure. So that's why it is the structure of the when doing the vaccine production, or when you think preparing vaccine, or when you think targeting the drug molecules, it is very important that the structure of this genome structure and what kind of mutation it happened is very important. Already people mentioning that some of the IITians and some national laboratories of India have studied these database submissions and compare the variations between these and COVID-19. They found about 153 variants in this small time, like December to June. If we count in the last 6 months, 6 to 7 months, we got so much variants of this. And in our state, actually, we have at this moment, every day we are testing, we find so many asymptomatic cases. They don't have any cough, they don't have any fever and all, but they are getting positive. They are showing positive in the test. And while doing the test, we go through actually RNAsp. RNAsp is the internal control to validate our assay. And MS2 phase is also the internal control to validate our technical procedures, isolation procedures. And EGene and RDRP and ORF1AB, these are the confirmatory genes to detect nCoV-19. So this is also vary from one kit to another kit. There are several kits we tested. We have used for RNA isolation also, we have several kits, you can see. And the basis for qPCR test is, it is all fluorophore dependent. Always the qPCR test is depends on the fluorophores, which is as the probes will be labeled with quencher and marked probes will be there. The reporter and quenchers will be there. So the reporter is will give a color, so that will give it as a signal actually. So that colors will be red, blue. We can choose which color. The fluoros will be based on the internal probe, which is reporter probe, which is varied in this detection procedures, and that will be detected by the machine, which is optical, and they have around 5 kinds of absorbent systems which detect these signals of amplification. Whereas in conventional methods, you are very aware of that agarose gel electrophoresis, and where you will do ethidium bromide staining, and simply we will gel run and we will see the amplification system. Here, the qualitative as well as quantitative detection will take place in, and also it is very sensitive than conventional PCR. Why? Because in conventional PCR, you can't detect a very single number or two number of copies. You can't detect, you can't visualize on the gel. Whereas in quantitative real-time PCR, you can also detect one single copy of the virus exist in the sample. Okay, generally we are collecting, we are all doing with throat swabs, which is collected by different field samples by doctors from ENT and doctors from eye hospital. They will collect and send it to us, and per day at least we will test 1000 to 1500 samples. Now we could be able to test every day in our lab at King George Hospital. And this is other parameters in diagnostics. You can see the qPCR diagnostic kit. This is BZI is the manufacturer. It is also a qualitative in vitro nucleic acid amplification assay. Here you can see only ORF AB gene. Actually, sometimes the target will be 4 in one kit, and in another kit the target will be 1 only. Actually, what we have observed is several false positives if the target is 1. Like in this kit, we need to test at least, if out of 100 samples, at least 30 repeats we usually do when we use these kits. And because these are all novel kits, still the R&D is going on which kit is the best to give 100% satisfactory results. So now we already have pick up some important kits from important companies, those who are giving 100% or at least 95 to 100% accuracy. That is also very important. As Dr. Vidyanidhi mentioned about the accuracy in reporting, we have a very hectic workflow that receiving sample and labeling the sample, and after labeling it will goes to the extraction, where the extraction sometimes the ICMR recommended to do pools, because we have enormous number of samples day by day. These days we are getting 1000, 2000, 3000, about 5000 samples in a day also we are getting, because our government has lifted the lockdown system, and travelers are coming from abroad by air, and also some trains are opened, and by road, buses, cars, all these people are coming. So that is the reason. Actually, still there are some quarantine people, around 2000 to 3000 people are in the quarantine in Visakhapatnam itself. You see, you just compare in others, because now the government of Andhra Pradesh facilitated every district headquarters as one laboratory they established in every district. So Srikakulam, when we start in King George Hospital Visakhapatnam, there we got samples from Srikakulam as well as Vizianagaram. Now they have their in a month they have their own labs at Srikakulam, and also at Vizianagaram in Maharaja College. Now in King George Hospital in East Godavari, now they are about to start in a few weeks. They will start a lab at VRDL lab by ICMR in medical college in Elur, actually. So like that, the workflow and samples, when samples and accuracy will be really very, very important. So that is a monitoring system. If one patient given a negative patient given a positive, it really reflects a lot in the community. And also one positive will be is become a negative, it also reflects the entire populatory and entire community contamination. Because if a person known as positive, if we give it as a negative, what will happen? He will go and spread entire his community, his family, or through family, his entire community will be disturbed like this. So we also find such kind of cases, but luckily we know we never have such kind of problems. In we have tested about 50,000 samples until from last 2 months. By today we tested 50,000 samples at King George Hospital, through our hands. And if you see here this ORF 1A, so by accuracy we ignored actually this kit. ORF 1AB is also common in another kit. We are now using applied biosystems where we are getting accuracy of 95% accuracy. And you can see using reverse transcription PCR. Actually, we have three steps, three-step PCR, and three-step PCR is a regular one, and we also have additional fourth step is reverse transcriptase, because it's an RNA virus. So our workflow started from isolation of RNA, so getting RNA, and we'll put it in the tube where we'll have the master mix, where it has all the probes and all kinds of target probes. And also in addition to the target, we will have reverse transcriptase enzyme, which initially in the first step itself, it converts the single stranded RNA into cDNA by using reverse transcriptase, where the reaction starts, PCR reaction, denaturation, annealing, and extension. So this is the workflow. And the persons who are tested are suspects and suspicious clustering cases should be in line with relevant guidelines of COVID-19 diagnosis and treatment released by local authorities. Actually, still going on. Actually, this process, entire process of testing and optimization of testing is already really comes to a good shape. But coming into the treatment parameters, see what they will treat if a person come with non-symptomatic. What doctor can how doctor can treat him, even don't know. There is no answer to anybody. Only doctor should answer to us, because what kind of medicine should give to the person who don't have any symptoms. Okay, just keep in mind. And if you think about COVID-19 diagnostics by qPCR, the kit is based on in vitro RT-PCR combining with fluorescent probing. Thus, just detailing about the technique here. The primers and the sequence-specific fluorescence probes were designed tailored to high conservative region in 2019 nCoV genome. The probes are oligonucleotide attached fluorophores at the 5' end with FAM as reporter and 3' end with quencher. In the meantime, specific primers and probes were developed as internal reference with fluorophore SWIC hex attached at 5' and as a reporter. Because in the multiplex, actually, if we have these different kinds of FAM, you can see FAM and WIC hex. These are all detectors, color detectors, which can emit the light. If the signal is strong, then it will give FAM, WIC, hex. So we know which probe is FAM, which probe is WIC, which probe is hex. For example, RNase P is FAM, WIC is our target, like as I mentioned, ORF1A, and other is hex is engine. Because when it should give different colors. Each probe should give different colors. Then only we can. It's a possibility to get the multiplex in a single vial, so that our time will be saved. In a single reaction, we can have the result immediately. But initially we struggled. Actually, some kits supplied individually, these FAM, VEX, and all. So individually means it kills a lot of time. In 2 runs only we can declare the results. But now that even the kits developed within a month, we have advancements in the kits, and we can give report in 1 hour, within 1.5 hours by the time we get the RNA sample. RNA extraction itself will take 1.5 hours. Then The workflow is 1.5 hour RNA extraction. PCR setup is about 30 minutes. Then PCR run is about 1.5 hours. So this is the total workflow. In this time, we can do at least 48 to 90 samples in this span of 3 to 4 hours. And in the diagnosis, during the PCR procedures, the DNA polymerase cleaves. This is the principle that how it amplifies. So the PCR polymerase cleaves the probe at 5' end and separates the reporter dye from the quencher dye when the probes hybridize to the target DNA. So this cleavage results in the fluorescent signal generated by the cleaved reporter dye, which is monitored real time by the PCR detection system. So monitoring the fluorescence intensities during real time allows the qualitative detection of 2019 nCoV in specimens. So if it is, we'll have reference control, and also the internal control. Every checkpoint will be there. You will have first checkpoint is internal control, and one more control is at RNA. We will add the phase RNA into the isolation procedure. So phase RNA-sp is the internal control. The sample is viable or not. We will know when RNA-sp is positive. Then we will know, okay, the sample is well, the sample is good. If it is RNA, if it is MS to phase, we will get the peak at MS to phase. So we can say RNA is fine. Then we will see the targets, the two targets. So ORF1A and also E gene. So these are all 4 targets. These 4, if amplified, will declare it as a positive. So if it is RNA-sp is positive and phase is positive and the other 2 are negative, means we declare negative. So because we will have cross check every phase, otherwise how we will declare the results. So that is the way we need to go for the workflow. And also cross check is very important in the diagnostics. So if, as I already mentioned, the report should be accurate 100%, not 99%. The results should be accurate 100%. And this is typical tests we will get in the kits. See, you can see 50 tests per kit we get, sometimes 100 tests. Nowadays we are getting 1000 tests. Initially 50 tests per kit we usually get. Now they are supplying 1000 tests per kit. So here you can see nCoV reaction makes. It is 1 ml 1 vial per 50 reactions we need to do. It is composed of reagent for amplification and probes and primers for target gene and internal reference, and enzyme mix, as I mentioned you, Taq polymerase, reverse transcriptase, and UDG. And nCoV positive control. We will also have positive control. I forgot to mention, we also have the, every time we keep one positive control, one negative control also as cross checks whether our reaction is working fine, because we don't have any, the reaction mix is active or not. So that we will know through positive control. And we also have blank control, whether any contamination of RNA or DNA from other source, which also could be prevented. And these are the presently available kits for nCoV-19 testing. For RNA extraction, we have MyLab nucleic acid extraction. This is also in collaboration with NIV, National Institute of Virology. They have really, it's indigenous company manufacturing, and the quality of the kits are very, very good for isolation. And Bath Biotech, it's a very new company, but we have very good experience in extractions with Bath Biotech. We really see, we have small eye on this Bath Biotech, but really it gives very accuracy as like Kaizen kits. It's international brand, but our Bath Biotech is really very good. And High Media is also a very good RNA extraction kit, which is used. We are all using regularly these MyLab, Bath Biotech, High Media kits, and also Kaizen. That is whatever the supply by the government, we are already optimized. Every batch we need to optimize. Whenever the kits comes, first at least we test 10 to 12 samples whether the kit is going in a proper way, because we also have such kind of problems when we start a new kit. So suddenly we'll have the very sometimes bad signals, so we can't interpret the results in such case. So that's why we always, whenever the new kit come, at least we need to optimize with at least 10 reactions or 5 to 6 reactions we need to optimize. If everything goes well, then we'll use in a bulk, because every time In every plate, we test about 100 samples in every plate, 96 well plate we use, so that 94 results. And sometimes if they are pools, 95 into 5. So we'll multiply with the 5, and each well contains the results of 5 individuals. And the PCR kits are Mylar PCR kit and BGI PCR kit. We ignored this BGI PCR kit because we are not recommending at all, because it's a Chinese kit. We don't like Chinese kits. And ABI PCR kits, so applied biosystem PCR kit. This is excellent kit. We have very in short span of time with ABI kit, you will get very immediate result. Within 1.5 hours, you can give the result of at least 95 individuals or 95 into 5 individuals. So these are the typical machines we are working, qPCR machines. You see the good genes, okay? Here, ABI Quant Studio, okay? These are all, we have these 2 machines in the laboratory at VRDL, Virology Laboratory at King George Hospital. And also recently we got 5plex. It is also available in our lab at Sjcw Molecular Diagnostics, as our students, even St. Joseph students, also have the exposure to this instrument. We show that we have a demo once they come to our lab for workshop. So I showed this instrument. The same instruments has been borrowed recently. 2 instruments has been supplied by ICMR to King George Hospital. There are 4 qPCR machines at a time. We can run about 4 into 96 samples at a time. We can run. And coming to that is the diagnostic part, entire diagnostic part I have explained in detail also. If you have any doubt after the completion of the lecture, you can ask some questions. And the present status of COVID-19. See, here actually in my last lecture, I delivered a lecture for Government College Rajahmundry. So where see the all over the globe, the confirmed cases are 42,58,666. The confirmed deaths 2,94,190, and the countries affected are 216 countries. See, now after a month, almost June 21st, you can see here, it is doubled in a month. The entire globe, 87,08,008. This is today, I got this information from WHO website, where you can see all updated information about this. You can see about the 46 lakhs. Here the deaths are 4,61,715 deaths, almost 10 times in a month. You can see 2,94,000, here 46 lakhs, almost doubled, I think, right? So see how it is transmitting. Even we have so many asymptomatic. You can see the deaths also. So that's why you keep in mind, don't spread corona to others in your community. If you have any symptoms, just stay at home. Use masks when you go into the public, when you go into the population. Okay, just use masks. Hand sanitizers you should have to use. Even asymptomatic also you will have deaths. And you can see in India, the status in India. See, Middle East, Southeast Asia, you can see 5,80,533. In that, see, India contributes a lot. 4,10,461 in India, and deaths are, see, 13,254 in last 24 hours. 15,413. Really, it is growing up rapidly. Really, it is going up very, very rapidly. All are listening? Hello.
Yes, I suppose so. Everyone are. I think they're very keenly listening.
Yeah. So that is the status. So it is in our hands. It is not in others' hands. So we need to have our own. See, we are in testing lab from last 2 months. We tested every 1 week whether we are positive or negative, because we are associated with samples. So even doctors are more at risk rather than us. Testing people are not at high risk. Only doctors will be at high risk. You might have seen in the newspapers, there are 4 to 5 forensic doctors, PG doctors at KGH got positive. So like that, so even they have more care, but we don't know how the virus affects us. So all precautions are, and also no gatherings. So group of people is, even you can move around the places, but you should not go for gatherings. That is the only preventive way. And you can see this is our team. I'm that there are two scientists, myself and Dr. Kalyani, my wife. You can see here, this is myself, my wife, and these are all these four members are our team members. And this guy is already our student. I trained him, and he also working with me at KGH. He is a fresher. He is from Krishna College, PG student, final year. He is also joined, and this girl is also Pravalika. She is from Bolaya College, PG, and this girl is Bhavani from Andhra University, but she working in another lab, and she also joined in our team. These guys are working very hard, because these are all in isolation team. So these four in isolation team, they always really isolation team people really I appreciate rather than.
Sir,
Pardon?
Dr. Jogeshwar, we couldn't see
Yeah.
the pictures. The slide didn't change. Either did you show it on the PPT or on the video? We couldn't see that. So
PPT only. PPT only.
It was not, the slide didn't change. The slide stuck to present status of COVID-19. Just check, sir.
Okay. Check. Yeah. Just, yeah. Yeah. Can you see now?
Yes, you can see that. Yes.
Yeah. So yeah, this is our group members, and there are 3 groups working. Actually, this is my group. I'm the team leader in this group, and Dr. Kalyani is associate team leader, and the other 4 are technical people. One guy, this guy is deputed from chest hospital. As you are aware that Dr. Vidyanidhi mentioned about chest and TB hospital, he is the senior microbiologist. Working there at chest and TB hospital. He deputed to KGH. He is in my team. And this girl is Pravalika. I think she graduated from St. Joseph's only. I think BSc at St. Joseph's, and MSc from Bulaya, I think. And this guy is from SV Krishna College. He recently completed project, his master's project under my guidance, and he also recruited. There, and total I placed 8 students of mine at KGH, because it's really tough to find the manpower, because there are no people have experience in working on RNA, and also they don't have experience on working on PCR, qPCR mainly. PCR, we gave the training, because generally people who are coming to our lab, they got acquainted with working with conventional PCR. The only thing is the analysis part is a bit difficult in working with real-time PCR. The reaction setting, everything is fine. But since it is a diagnostic, so we need very experienced people. That is the reason it's tough to find the people who work.
Is your video in off mode? We can't see you actually, though of course we need to see the PPT, but we would have loved to see you parallelly also. Is your video in off mode?
My video, okay, just yeah, it is in off mode. It started.
Now that you've almost finished.
Yeah, so
very good. It's almost like a frontline team, Dr. Jogeshwar.
Yeah.
Good.
Yeah, ma'am. Because the extraction is because what happened in labeling parameter, as Dr. Vidya mentioned in his talk, the labeling parameter is very, very important, because we are not handling 1 or 2 samples. We are handling thousands of samples, hundreds of thousands of samples. So in such case, if 1 sample labeling is also missed, the accuracy of the testing will be questioned.
Yes.
So that is the main issue we faced, and really that's why the sincerity and also the presence of mind and everything is very, very important in diagnostics rather than in research. Okay, if anything goes wrong, you can repeat, but here we don't have
chance
once you delivered a report.
Yes,
So
true.
that is the issue to deal up. Really, in this last 2 months, we never have a one single mistake in our hands. So really, it's a God's blessings, and really we all really when we go to join and when we go to work at KGH, really we got afraid. Why? So he asked, the HOD of microbiology asked me and called me, and he asked me, why can't you come and work in this and lead the team? So really that question makes me afraid.
Yeah,
That question makes me afraid that day, because we don't know about this virus, how it is going on spreading,
yes,
and even
very
if we go there, yeah. So we take the risk that time actually. So he also appreciated about it, and our district collector also, we have meetings, several meetings with district
very
collector,
good.
and also we have joint commissioner is there for this. He is the leading officer working on this, and he also appreciated because by the time before we go to KGH, only they could able to test 25 or 30 samples a day. Now
Yes.
after we go there and we build up the team, now we can able to test 1000 to 1500 a day. So that is why in a day 1500 means in a month we can finish 30,000 samples.
Yes, yes.
So that's the reason. The resources is very important, and also I recommend all the students actually, they should have to, because now a common man also know what is PCR.
Yes.
But the thing is, yeah, okay, I understand the quality of your education. I know about the quality of St. Joseph's College for Women.
Thank you
I
so
know the
much.
quality. Really, I have several students from your college, and come for the workshops or come for training purpose. Even people in the participants also might be listening. I think they're really sharp.
Very,
What I have seen, even
we are
some
really
PG
honored.
students.
Thank you so much for
Yeah,
the
yeah.
confidence.
Yeah, because I always associated, I take the classes, so I ask the questions every time. My way of training is question bank. I ask
Yes.
the questions only when student responds, I proceed further. So
Yes.
that's why I study every student's mind. And what I realized is, uh, actually we need to strengthen our biotechnology. uh, because see, hardly I got a call yesterday, so they need resources again to build up lab at Elur.
True.
So I can't recommend one single guy for that position, because
Yes.
there are no people trained for real-time PCR or RNA extractions like that.
Yes.
So
So we have a few questions, Dr.
that is
Jogeshwar.
very important. I think you people are following that. Yeah.
Thank you so much. If you disable
Yeah.
your screen, then so that you can disable screen sharing, so that we'll see you in a larger this thing in the video. There are a number of questions in the QA box, and Dr. Veerbrahman will ask them. But before that, I see two hands raised. Can I allow them to talk? Two hands, and then Dr. Veerbrahman will go on to the Q&A box, which is the same. There's one question by Bandi Tarun. I will allow him to talk. Mr. Bandi Tarun, if you unmute your audio, you may ask the question to Dr. Jogeshwar. And there's one more hand raised, and after that, Dr. Veerbrahman will ask the questions from the Q&A box. Dr. Jogeshwar, is that okay? Is that okay, Dr. Jogeshwar?
Yeah, madam. Yeah.
Yes, yes. Bandi Tarun, are you coming in? Okay. Chitturi Gnana Prasuna is ready. I'm going to allow her to talk. She's ready. Prasuna, are you asking your question? If you start, otherwise we'll ask Veerbrahman to go on with his questions. We don't want to waste time. Are both of you coming in, or?
Yeah.
We are allowed them. We have allowed them. We've asked them to unmute, but.
Yeah, madam. Actually, meantime, actually there are 5 Q&A here. I'm going through that. I'm reading out. Actually, Parmila Kopalle, how does the asymptomatic person spread the disease? Actually, symptomatic and asymptomatic, the same way it spreads. That means the only way is physical communication, or through only the what we can say it as a through mouth only.
Yes.
Majority of this virus spreads through mouth, spreads through whatever the droplets from your mouth, and also sneezing. And if you have that infection, even it is asymptomatic, if you sneeze and if you contaminate, or if you have through that only it will spread, irrespective of asymptomatic or symptomatic. And when Dr. Bindia, hi Bindia, I know Bindia very well. So with rising asymptomatic cases too, I request you, sir, to advise higher authorities to kindly go back to lockdown, since you are playing a prime role in detection studies. No question on talk. Actually, the thing is, why our government has that main thing is, see, our all systems are getting disturbed. That is, I recommend our population, our people to be in their own zones. Actually, they should have their own limits. They should keep their limits. If we cannot ask the government to stop the entire things, because already we have 2 months we faced, and everything economy has disturbed, and also you can see the death rate. Generally, the death rate of if you see general diseases death rate, it is also almost similar kind of death rate. Even though more are getting infected, more people are getting infected, but our Indian immunity system, as people are saying, and this is more in the debate, more in the discussions, that the death rate is low. That is the reason it is ignorable. 2.5% is the death rate. So death rate below 5% or 10% will be acceptable. So that is the reason government has lifted lockdown. Government lifted lockdown means it is not the freedom to give to the people. So people should also have the awareness. In the last 2 months, what have done? They only go for limited things, unpurposeful things, not general gatherings. So I don't recommend general gatherings. And instead, if we do lockdown 3 or 2, it will affect our really finance system, everybody's finance system, not only the government's finance system. It's the people's finance system. And also, how we will get the food if everybody shut down? They don't produce the grains this year. So how we will get the food? So these are all the things. These are all we need to, as a human being, we need to think these things ourselves. And I think, as Modi said, there are no more lockdowns. And another question by Dr. Bindia. Madam, we have time. Can I go through like this, or
maybe you just have time for 2 questions, Dr. Jagdish.
okay, madam.
Somebody
are online. Somebody are online.
Yes, please.
Yeah, madam. Yeah,
go through whichever you feel
madam.
most relevant. No problem, please.
Are the kits you are getting from Indian made or other countries? Can you specify from where you are getting kits to identify COVID? Actually, now, very firstly, when we start our work first week, we used the Chinese kits only, BZA. Now we stopped. We are now only using our indigenous kits, like MyLab kits, and also some are from other countries. ABI is from other countries. And Sai Sindhu, why we are not using BGI PCR kits? BGI PCR kits, actually there are so many false positives. So that is the reason, because only one target is there. That is in only when we analyze a single target, if we get some peak, so we can't judge. That is the reason. So multi targets will have better reporting parameter. So that's why we stopped using BGI kits. And Dr. Sunita, Glenmark and Hetero launched medicines for COVID. Is there or useful any information about those? Yeah, these are also common drugs. We don't have any clinical data on that. I expected this question, actually. So good question, Sunita. And actually, we don't have any clinical data, so we can't talk about it. But it is an antiviral drug. It is common antiviral drug. So in something is better than nothing drug. That's it I can say at this moment, because we don't have any clinical evidence. And can coronavirus survive on surfaces? No, coronavirus cannot survive on normal surfaces. It immediately die. Only it can. It need a cell. So that's why it penetrates very rapidly. Once it is enter, it can survive. So outside surfaces, it can't survive longer times. Maybe shorter times it can survive, but longer times it can't survive. That's it. Thank you very much, madam.
So there's one question.
Yeah,
the last
madam.
question, sir. What kind of mask? Prasuna is asking, what kind of mask protects us from COVID-19? Because there are so many masks.
Actually, yeah, madam. Yeah, madam. As we are working in the laboratory, diagnostic laboratory, we are just using even surgical mask also. We use it in sometimes in testing also. Sometimes we have lag of these N95 masks. So N95 is routinely using masks. If you can also avoid protruding of this virus through surgical mask also. But I recommend N95 mask only.
Oh, then good. I thought, oh, these ordinary masks are okay. Okay, I'll also buy N95. Okay. Not the designer masks, no? What you're saying?
Madam, even surgical mask also is also we don't have problem. Surgical mask, but we almost use in laboratory N95 masks only.
Okay. If you can disable your screen sharing, then Dr. Veerabrahmachari also can log in and share. If you disable your screen sharing, you can also be in the.
Yeah, madam. Yeah.
How are you having a little free time today out of your busy schedule?
Actually, yesterday after 3 shifts, we'll have one 12 hours phase, vacant phase. Yeah, after 3 shifts. So again, we have night shift today, 10 to morning 8. Today we have 10 to morning 8. It's fortunate that I am in this shift actually. Luckily in this. Yeah, that's why actually that is the reason I could not that mock day you have done mock that day I was not able to come actually. Yeah, madam.
But I must say, Dr. Jogeshwar, a big thanks for you from St. Joseph's College on behalf of our principal, Dr. Sister Sjcw, the management. We are very happy for all the compliments that you have showered on our students and on the college. And I must say, let God bless you and your team with lots of health. You're working in a very risky zone, I know, with lots of energy and enthusiasm. I must say, I salute you. Hats off to you.
Yeah, ma'am. Thank you, ma'am. Thank you, ma'am. Thank you very much.
May God bless you, right? All of you are much younger than me.
Thank you.
I've just retired. So may all of you be more blessed. And please take care of yourself also in that. Thank you very much. See you again sometime, right? Thank you. So we now come to the last third session of the wonderful biotech webinar. And we already have our third resource person ready. And I'm wondering if, yes, we are very happy to have Dr. Veerabrahmachari, again, a very young, very smart scientist who has a big record of achievements. And we are ready for the third session. Once again, he will take us on another beautiful session in the biotech webinar. It now gives me great pleasure to invite Dr. D. Manishri, Assistant Professor from the Department of Biotechnology, to introduce Dr. P. Veerabrahmachari. Over to Dr. Manishri.
Thank you, ma'am. Dear participants and our esteemed resource speaker, I, Dr. Manushree Dekonda, Assistant Professor from the Department of Biotechnology, St. Joseph's College for Women, deem it my privilege to introduce a very vibrant speaker, Dr. P. Veerabrahmachari, Assistant Professor, Senior Scale in the Department of Biotechnology at Krishna University, Machilipatnam, South India. With a PhD in microbiology from Goa University, India, Dr. Veerabrahmachari worked as postdoctoral research scientist at the Bacterial Pathogenesis Laboratory, Queensland Institute of Medical Research, Brisbane, Australia, and DBT postdoctoral fellow at the Department of Microbiology and Cell Biology, Indian Institute of Science, Bangalore. He published more than 120 research articles, including 5 international books in prestigious peer-reviewed Scopus Index journals, and presented 50 abstracts at various national and international conferences. He is a reviewer and on the editorial board member for a number of national and international journals. He is a member of many international scientific societies and organizations. Most importantly, Indian Science Congress and Society of Biological Chemists, India. He successfully completed 2 major research projects with major interest in biochemical mechanism of bacterial biofilm formations and translational cancer research, among others. He has been honored by many international awards, notable among them being the Young Scientist Travel Fellowship for the year 2007 from the DST Government of India for attending 17th Lansfield International Symposium at Porto Heli, Greece. Andhra Pradesh State Best Scientist Award for the year 2017, as well as Dr. V. Ramalingaswamy Memorial Award for the Biomedical Sciences 2019 by Science City of Andhra Pradesh. With a Government of India Young Scientist Award way back in 2011, it is no wonder that Dr. Veerabrahmachari has 2 patents in his chosen field. We welcome Dr. Veerabrahmachari and feel privileged to have him as our final resource speaker for this one-day national webinar in biotechnology. Now I invite Dr. Veerabrahmachari to take over this session. Over to Dr. Veerabrahmachari.
Sir, please unmute, sir. Sir, mic unmute, sir.
Yeah.
Okay, sir.
Yeah.
Okay, sir.
Okay. Is my voice audible, all of you? Yeah.
Yes, sir. Yes, sir. It's audible.
Yes, it's audible. Dr. Veerabrahmachari, you can continue.
Okay. A very good evening, all of you, and most respected greetings to the principal, the chairman, the secretary, the organizing
Voice
team.
is maybe a little louder, sir. Can you raise your voice a little louder? Yeah. Thank you.
Is it okay? Is it okay, madam?
Yes, it's okay. Dr.
Yeah.
Veerabrahmachari.
So I hope it's the end of the day. I just would like to ask a question. Like, we all have a patience to listen to my talk for another 1.5 hours. Are you all set, madam? Are you all set to listen to my talk for another 1.5 hours? Is my voice audible, madam?
Yes, your voice is audible, sir.
Audible. Okay, fine. So greetings to the department, the principal, the chairman, the organizing team, the convener, the teaching fraternity of the college and the Department of Biotechnology. I deem it a privilege and honor for inviting me to deliver a talk on this auspicious occasion on this evening. And the 2 speakers who spoke in this evening, in this afternoon, are my good friends and my classmates. Dr. Vidyanidhi is my good friend, and Dr. Yogeshwar is my classmate during my master's. And today I would like to talk on evolution of SARS-CoV-2 infection, pathogenesis, immune evasion, and vaccine strategies. I'm pretty sure Dr. Jogeshwar must have already given some kind of some highlights on the diagnostic part of the SARS-CoV-2 and its importance in the healthcare industry and medicine by and large. And let me talk about, so are you all audible, madam? Is my voice audible? Hello? 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 Hello?
Sir, it's okay, sir.
I can hear some background voice from somewhere. Can you ask them to unmute all of them? I can hear some background voice. Can you ask everyone to unmute? Hello? Hello? Okay, shall I go ahead?
Yes, sir.
Yes, sir, where it is coming from. But
Yeah.
none of us are.
There is some voice coming up. Hello?
Can you hear any other voices?
No, I cannot hear.
Please.
It's fine now. Shall I go ahead, madam? So please make sure there is no disturbance because I'm a very sensitive speaker. I do not like a background voice, madam. Please take care of that. Yeah. Yeah, I can still hear some music sound coming from one of the participants' background voice. I think someone is playing music.
Sir, here all participants are mute, sir, because of you can see your surroundings.
No. I'm fine. I'm sitting in a very contained room, closed room. Okay, fine. Shall I go ahead, Veerabrahma? Shall I go ahead?
Yes, sir, you can continue.
Okay, fine. I am speaking on infection pathogenesis, immune evasion, and vaccine strategies. And this is the image which I would like to show you how there is some song playing. This is disturbing me. Someone's song is playing. Please look into that. Anyasri, please look into that. Someone's song is playing.
Now it's okay, sir.
Now it's okay.
Okay.
Please ask the moderator to mute all the voices. Yeah.
Okay.
Because,
okay, sure.
Because this is my start up with the talk, and I definitely would like a pin drop silence. Yeah. Fine. Shall I go ahead? Yeah, it's fine. Thank you. So this is the image depicted on my cover slide. Is nothing but the COVID virus, SARS-CoV-2, and the whole theme of the talk is there in my first slide. That I'm talking on the immune evasion and vaccine strategies and the target organ, that is the lungs, which is located here. Okay, so we'll talk much deeper in right from beginning onwards. What is the history onwards to the evolution, the infection, the pathogenesis, immune evasion, the technologies involved in designing vaccines, the platforms used in the current world to combat the disease. Okay, so when you look into this slide, especially, the world has really experienced a kind of a public health emergency in the name of pandemic and in the name of outbreak, in the name of quarantine, in the name of isolations, in the name of several whatnot. Okay, so CDC, Center for Disease Control, Georgia, Atlanta, and WHO has given very clean regulations on the travel advisories of the people traveling abroad across the world and coming to several places. Yeah. And there are several regulations given by several advisory boards, by health and medical emergencies as declared throughout the world. And even till today, we are also experiencing the same kind of outbreak situations, and we are actually feeling it. And also recently we have seen in so many televisions that so many disturbing videos we have seen, so many laborers coming up from their workplaces to traveling back to their home places, and meanwhile several people have died in between, and they have lost their lives. And even in the hospitals we have seen so many emergencies, so many casualties, people dying due to severe acute respiratory disease syndromes and acute respiratory disease syndromes. I'm pretty sure we may have to face it we may have to take it as a challenge and live it and live with it, and definitely we may have to look out for some very much alternatives to live with it, and definitely live up to the standards of the sanitary conditions and the healthcare precautions we need to take. So if we talk about the history of pandemics out here, the pandemics what we are talking about today, about novel coronavirus, is COVID-19. It's just a small dot compared to the previously occurred pandemics. Look at here, okay? Yellow fever and Russian flu, Spanish flu. Look at the Spanish flu, the biggest, 40 to 50 million. We are now talking about 8 to 9 million cases, infected people across the world. And look at this, the plague, and look at the cholera, the blue one, the 1 million, and 600,000, nearly great plagues, and so many plagues have really hit the deadlines of hit history of pandemics. And look at here, this 56 million people died due to smallpox, and bubonic plague, nearly 200 million. It's quite larger compared to your Spanish flu, and 30 to 50 million even the plague. So many plagues have really hit the history of pandemics. Okay, so we are very small dot. Okay, and even SARS is also here, and Ebola is also here, MERS is also here. Okay, swine flu, which has come up recently, is 200,000. Okay, so this is one great thing which I would like to show, how the humans have spread across the world, and infectious disease have been a constant companion. So it's now again a companion to all of us, and this is the most deadly pandemic, okay, right from beginning onwards. And let me talk about, I think you all must be heard about what is called tissue tropism. I'll talk about that in the later stage. Okay, tissue tropism in the sense, it is basically an animal virus, and basically a zoonotic transfer has occurred from animal viruses to human beings. So several animals are really acted as disease reservoirs and intermediate hosts. So it occurred in 5 stages in terms of this virus, especially. This virus is basically a coronavirus, and here, if you look into second stage, the bat is the actual culprit. I'll talk about the bat at the later stage. Okay, even Ebola and SARS and MERS, for all of them, the bat is a major culprit. And here, there's a species barrier. There's a species jump that has occurred from bats to the animals like civet cats, swines, camels. Okay, it has gone to all 3 things. Again, there is a species jump, species barrier from animals to human beings. And here, there's a transmission of humans that between humans and humans. Okay, now here, the virus is really transmitted very badly across the world and across the countries and across the states, cities and towns, and even to small villages. Okay, now it is in the dissemination stage, in stage 5. Okay, it is aided by increasingly mobile populations, and definitely we are really experiencing the kind of a heat coming out from the local transmissions and the worldwide transmission across the globe. So, well, if you talk about this virus as a whole, coronavirus basically belongs to Coronaviridae. Okay, it comes under four families: alpha corona, beta corona, and gamma corona, delta corona. And the corona which has infected the human beings is beta corona, lineage A, lineage B, C, and D. Okay, so we are basically coming in lineage A because lineage B has come from MERS and SARS-CoV-1. Okay, so if you look into here, there is the bat as a major reservoir. Okay, and there is a lot of intraspecies transmission that has occurred from bats to another bats. And then interspecies transmission occurred from bats to other animals like cats, civets, swines, camels, and so many other animals have really got, even macaques, chimpanzees also have infected. Okay, so from there it has moved to transmission of human beings. Now people are scared if from human beings, if it jumps from the same strain, if it jumps from other species and reverts back to human beings, okay, and this time I'm pretty sure it's not going to be 8 or 10 millions, it's going to be more than 50, 60 millions. People will die just like that within seconds. Okay, so enough of mutations have taken place now, and probably we need to really look into, we need to really stop the virus mutating at one particular place. So we'll talk about that. And if we look into the SARS and MERS, okay, so MERS and SARS And one more is SARS-CoV-2 and SARS-CoV-1. Okay, all three are cousins. They belong to same virus, coronavirus family, and all of them are closely related to bat strains of corona, and they transmitted other secondary infection species. Okay, so the SARS originally associated with the wet market in Guangzhou. Okay, which has come in 2002. The outbreak has recently come from from a Guangzhou market in 2002. And if you look into the phylogenetic analysis, look at here, the SARS, the initial one which came up in 2002. Okay, so if you look into all the sequences are pertaining to the bat, okay, the SARS-CoV-2 is somewhere in between the red one, okay, and MERS is somewhere in between here. MERS has come from the camel. We will talk about that, okay. So if you look into the relation between SARS-CoV-1 and CoV-2, okay, there is 80% similarity in the genome, and we are talking about 0.5% similarity with human beings and chimpanzees. We are just 0.5 to 1% dissimilar with chimpanzees. We are 98% similar with the chimpanzees, okay? But the SARS-CoV-1 and CoV-2 are 20% dissimilar, okay? So we can imagine what must have taken place. There must be a very large fragment which must have either deleted or inserted from the wild type viruses, especially from pangolins or from bats, okay? So if you look at here, bats as a reservoir for emerging viruses, okay? Why bats? Why no other animals are getting infected? Why only bats? Even in Kerala recently, we have seen a recent outbreak of viruses like Nipah virus, which came up in Kerala. Okay? Even that has come through bats. Okay, the great thing is at least they have pointed out that it has come from bats. Bats eating the fruits and people eating the fruits in turn has acquired the infection. Because bats are very diverse. I'm pretty sure. Okay, you must be amazed by listening this. Okay, bats can acquire nearly 600 viruses at a time and become asymptomatic. Okay, because of their food choices, they have diversified food choices. They are colonial or solitary in nature. The population structure, ability to fly. While flying, it has a ability to disseminate the disease from one place to another place. Seasonal migration and daily movement patterns and lifespan is pretty high compared to other avians. Okay, avian mammals. Roosting behaviors, ability to ecolate. Okay. Echolocation is one of the things. They can echolocate to any new place pretty good, and virus susceptibility is also pretty high compared to other animals. Okay, so virus can really take up so many viral diseases can be acquired in bats, especially rabies, Lissa, influenza A, Hendra, Nipah, Corona, Japanese encephalitis, even chikungunya also will come from some particular bats, okay? And if you look into the bats, because we feel that bats are very important in pollination, help controlling the insects, reseed the cut forests and pollinating the plants, and their even their guano, their feces are used as fertilizers, and the models for sonar systems like echolocations. They have the very good sonar systems. Okay, there are so many advantages as well. So why are the bats reservoir to so many deadly viruses? See, a very peculiar thing you must not have heard and known anywhere in your whole life. Okay, they have a difference in their innate immunity. When you compare other animals and human beings, their innate immunity is entirely different, and the constitutive interferon production. Okay, so throughout the life, because it's harboring 600 viruses, constitutively the bat will be producing interferon in the body, in the cells. Okay, decreased or absent expression of pyrine or HIN domain proteins. These are the responsible for innate immune response proteins in viral co-evolution and part of bat gut virome. Okay, so virus has co-evolved along with the gut virome of the bat. Okay, so I will rather say that the bat has adapted, and the bat has adapted the virus, and virus has attenuated the cells or cell lines in the bat, and over a period of a time, both the receptors has become co-evolved, and now I can rather say that all the bats are in the mode of asymptomatic phase. The flight results in the increased reactive oxygen species. And throughout the time, okay, while in the flight, while flying, the increased reactive oxygen production and the body temperature will shoot up pretty high, and that body temperature will be very much interested to grow any kind of a virus, and especially SARS-CoV-2. And some people have recently argued that high temperatures will kill the virus. I'm pretty sure that logic is not at all good. And we have also experienced 45 to 50 degrees Celsius in Andhra Pradesh, and still we have seen so many pandemic cases, so many SARS-CoV cases. Okay, so what I feel in the bats, whatever is happening in the bats, I'm pretty sure that also happens in human body as well, because increase in the flight, increase in the anxiety, the stress, okay, reactive oxygen species production will be increased, and likewise, the body temperature will be shooted, and it will be very much suited for the virus to grow inside the bats and also human beings. So viruses may also have adapted to the higher temperatures. Look at here. So the adaptation to the higher temperatures is also very good model system for the virus to grow in the bats. So that has made the bat to transmit the virus in pretty well when compared to other intermediate hosts or reservoirs. Okay. And there are three reasons why the bat don't get sick with the new coronavirus. The thing is, I talked about the evolution. See, they have co-evolved with it in terms of receptors. Okay, so unlike in human beings, immune system fights the viral infections pretty well. They have got very constitutive immunointerferon productions, and they do not overreact with them. And the funniest part in bats is the deadly hyperinflammation. Okay. That does not occur in bats, okay, unlike that happens in the human beings. Okay, we'll talk about the hyperinflammation. If you remember what is hyperinflammation, then you remember about cytokine storm, we remember about hypoxia, so we'll talk about that at a later stage. Okay, so so virus has become less lethal to the original host. I told you before, the virus has become attenuated to this host, and it does not infect the bat cell lines anymore, and now the bat has come to a stage that it can only transmit the disease, just like the swines, the pigs, the pigs also can harbor nearly 1,000 viruses at a time, and no viruses can infect the pig, even Japanese encephalitis or chikungunya. There's so many viruses coming up from swine flu, H1N1, H3N7, all the things. The intermediate host reservoir is the pig actually. Otherwise, it must have come from birds, but the reservoir, the holding reservoir is a pig. Okay, so if you look into bat coronavirus, okay, so I told you these three people, the three are the killer pandemic enemies. They are the cousins, okay, they belong to same family, coronavirus. And 2002, it has come up in, okay, SARS-CoV-1. It has initially come, the initial actual thing is the bat, okay, the transmitter is bat. It has come to civet cats, and here COVID-2, the transmitter is bat, and the reservoir is pangolin, and here MERS. From bat, it has gone to camels, and camels again. All the three of them now has spilled over to human beings, okay, in various times, like 2002, 2012, 2019. So this is a spillover time, okay. So I think all three animals have spilled over beings on the human beings. Look at here. Look at the deaths cases, and number of cases occurred due to COVID-1 was pretty low, and MERS is also pretty low, but the R0 reproductive number is pretty high compared to SARS-CoV-1. Okay, MERS is much more infectious than SARS. Okay, which is known as MERS is nothing but a Middle Eastern respiratory syndrome virus. It has come up in Middle East, Gulf region. So many camels were infected, and from there it has come to human beings, and this is now SARS-CoV-2 or COVID-19. Okay. So as on today, it is 8.9 million infectious cases, okay, nearly 3 million death cases across the world. Okay, look at the kind of a pandemic what we are experiencing. Okay, then if you look into here, the origin for SARS-CoV-1, I told you it's bat, and intermediate host is civet cat, and from there it's gone to human beings, and MERS, same as the bat, same for all the three viruses, all the three cousins. Okay, the origin of the virus is bats, intermediate is camel, and here they are really saying whether it is pangolin or directly from bat. People say that Wuhan, some people eat this pangolin as a delicious food, as a tradition. Okay, so that is how it has harbored. Even this immune system is very peculiar compared to other animals. Okay, the whole genome, when aligned with the genomes of other viruses, bat COVID RAT G13 region. Okay, this is known as Rhinolophus affinis species. Okay, it's known as horseshoe bat. Okay, of Yunnan province with 96% similarity. COVID-19 virus was likely emerged from recombination of viral genes across. So the gene must have recombined from all these three species. Okay, I'm pretty sure some larger fragment has been picked up from one species while a species jump has occurred. Okay, and that larger fragment is being replaced in the human COVID virus. Okay, so that is the major reason. And if you look into the pangolins, may have joined as an intermediate host. Okay, we are talking about this pangolin here. Okay, this pangolin COVID whole genome had 91.02 similarity with the SARS-CoV-2 and 90.5 similarity with the bat COVID RAT G13. Okay, this is a recently 2020 paper. Okay, so that means we have to believe that this is much more closer compared to the bat COVID 90.5. Okay, so the emergence of SARS-CoV-2 through recombination and strong purifying selection. So there is a strong recombination and purifying selection saying that yes, the gene, the inserted gene has aptly placed in one particular place, and recombination and mutation and has occurred, and then that has really made this virus much more stronger in terms of the selection in the species. Okay, and SARS-CoV-2's entire receptor binding motif, RBM, was introduced through recombination with the coronavirus from pangolins, possibly a critical step in the evolution of SARS-CoV-2's ability to infect human beings. So this is the major thing. The receptor binding motif must have strengthened during the insertion or during the recombination and during this very strong selection. Okay, so the potential origin of SARS-CoV-2, if you look into here, okay, basically, I'm talking about the SARS-CoV. Humans are these are the zoonotic species which generally infect the human beings. Okay, and most preferably, only these 4 species were known till now to infect human beings, and I don't know, these 4 are not reported, and finally there is some recombination mutation occurred between 4 species. Okay, and then come to the human beings, and from human beings to human beings. Okay, so human to human transmission, and now it is and all these animals have become asymptomatic, and now the cycle is running around between only only human beings. Okay, so if you look into red arrow lines shows the mutation, and the purifying selection is pretty stronger compared to other things. Okay, when you talk about the single line SARS-CoV-2, okay, this is moderate. Okay, and when you talk about SARS-CoV-2 from here to pangolin to human beings, this transmission is very high. Okay, so reproductive number is pretty high compared to this one. And when you talk about the merged transmission, okay, it's much lower. Blue is much lower, is moderate, and is pretty higher. Okay, so the human to human transmission and this transmission, the dissemination of infectious virion particles from one person to other person is pretty higher compared to these 2 cousins. Okay, so when you look at the genomes of these 3 people, okay, these 2, the genome is 80% similar. Okay, this is much more dissimilar, at least 35% dissimilar. Okay, so when you look them, the phylogenetic network of 130 SARS-CoV-2 genomes, 160 SARS-CoV-2 genomes are picked up and recently come up in 2020 PNAS paper. Okay, they have observed that node A, this yellow is a node A. Look at the node A, how it has originated from here. This is a root of thing, and from here how it has moved from. Okay, is a root cluster obtained from the bat, and from there it has moved to. Okay, 1-point mutation and 2-point mutations and 3-point. So a single mutation in 1 point will become a one particular dendrogram. Okay, one particular population, and there is one. This yellow, you can see the yellow everywhere. Okay, yellow is come from China. Okay, And I think bat, you forget about bat. If you look at the East Asia, okay, it's pretty less. Yellow is much more, and the transmission of the bat virus across 160 genomes is pretty higher compared to other bat genomes. Look at 160 SARS-CoV-2 genomes, okay? So yellow is everywhere, okay? So this is what has happened. And if you look at here, the potential origin, they're made into 3 clusters. 1, 2, and 3 are the major clusters. Okay, these are the clusters. We can call them as an ancestral type. Okay, so they're closest to coronavirus found in the bats and pangolins. Okay, so A is the one which has come from bats and the pangolins. Okay, and B is the one which has come from Wuhan. Okay, from cluster A via 2 mutations. Okay, this, if you look into here, there are 2 big large mutations occurred here in cluster B. And cluster C, I'm talking about cluster B here, okay? This cluster B is very much packed, and this has come from Wuhan, okay? And cluster C is a daughter of cluster B. So this cluster B, cluster C is a daughter of cluster B, okay, which came from Wuhan, okay? So one mutation different from the parent variation, okay, has spread to Europe via Singapore, okay? So if you look into the large part perspective of it, okay, so I think you are giving me very less time to speak, madam. I understand. I got a message that yeah.
Please pardon me, and I ask your apologies, sir. There was some technical error that at the time exactly we will stop at 5. So I'm wondering. I know my tongue is tied to ask you, but let's see how you can manage it, sir. Give us just 5 minutes before if you end. Then we will.
Usually, when I deliver a talk, I don't look into, because I'm just an initial
yes.
Probably, yeah. So
please go on, sir. Please go on. Please go on.
We'll see and how we'll go on, and you can stop me at wherever.
Please go on, sir. We will we'll quickly schedule another meeting. Not to worry. Please go on.
Okay. So if you look into the evolution part of it, okay, this is SARS-CoV-2, which has come up in 2002, and if you look at the pangolin, okay, the COVID-2, which is very, very close to bat RAT G13 region, this lineage is leading to RAT G13 apparently lost the specialized receptor binding. So this has lost the receptor binding domain that helps the SARS-CoV-2 infect the human cells. Okay, so I'm pretty sure there's something must have happened here. Very strong purifying selection, or very strong mutation has occurred between RAT G13 region of bat COVID virus and the SARS-CoV virus, and that particular region has been replaced and placed in the human cell lines, which is infecting them. So when you look into the past, present, and the future of coronavirus, okay, the evolutionary thing, okay. So what evolution may have led to SARS-CoV-2's ability to spread? The ability to spread of COVID-2 is very, very large. And how is SARS-CoV-2 evolving during the pandemic, and what future evolution could be problematic? I told you, if at all, if any such species jumping or species barrier may occur in the next species, okay, and again comes back to human beings, I'm pretty sure this time it's going to be 50 million, not 8 to 9 million. Okay, so we must be prepared for that. So I'm talking about the spike protein evolution. Okay, till now we talked about the evolution of COVID-2, and let me talk about the spike protein evolution that must have led to enhanced interaction between the human ACE2 angiotensin converting enzyme receptor, which is there in the human beings. Okay, so if you look into here, there are 4 different places the mutations have occurred in the bat, and again the 4 different mutations have occurred in the human beings. And again, 4 mutations are very strong mutations, very strong 4 mutations occurred in the point mutations occurred in the bat, and that region has carried to human beings, and that region has carried to both, just where both human beings and pangolins are sharing them. So a recombination that enhances the S interaction. S is a spike protein interaction with the human ACE2. Look at here. So whenever you see this, okay, it's much more very close to bat, and it's still, there's a big doubt is arising that whether it is come through pangolin as a reservoir or intermediate host, or bat as a reservoir host. Okay, and people are predicting that some amount of a yellow orange gene, okay, some large gene has picked up and put in the SARS-CoV-2 with this insertion and recombination from pangolin gene. Okay, and anyhow, most part of the gene is belongs to RAT G13, and because now it is a hybrid of both bat virus and pangolin virus, so this large gene of nearly 7 to 10 kb fragment is inserted here, and that's the reason why it is really not listening to any of the public health perspective, and it is really causing a devastating effects across the world. I'm pretty sure we may have to experience some more casualties in terms of millions at the end of August or September. Okay, so new study finds, it's come up in 2020 Scientific Advances, finds that recombination is very, very common among the strains of coronavirus. Look at here, that recombination is occurring only between the B, C, D, and E regions of the spike protein, only 4 regions, okay? So you take any genome and do the phylogenetic analysis, okay? It is showing the evolutionarily, it is linked up with either pangolin or either bat or human beings, okay? So none of the 4 structures are similar. Okay, you take any genome of spike and see the phylogenies, okay? They are yielding a different tree structures, indicating that these different segments of the genome have different evolutionary histories, an indication of a very strong evolution, a very strong recombination and a purifying selection across, especially the spike region of the COVID-2. So even if you see here, there are 4 different species, and okay, the mutations have occurred in 4 different places. Okay, look at here, this is a different place, even this is a different place. Okay, so if you look at the locations, how these people are disseminating from one location to another location, this virus has got a capability to move from one location to another location pretty well. Okay, when compared to other viruses, this kind of a location movement, because even that is also has a very good flight, moving from one place to another, echolocations, okay? So the virus has got an ability to transfer very quickly to the farthest place through bats as a reservoir host or a disseminating host. So if you map them in the phylogenics, okay? So each mutation is like a breadcrumb left behind the path. Look at here. So all 4 will fall into 4 different clusters and 4 different clades. So obviously, the entire phylogenetics will change based upon the structural mutations on one particular spike protein. So if you look into the COVID-2, if you look into the genome variations, okay, they have taken so many genomes and they started mapping them, okay, sequencing and pulling them together. And we see this SARS-CoV-2 is behaving very differently when compared to other genes, because I told you very, very strong recombinations occurred in between them. Okay, so even same which I showed you in the before slide, this has come up in recently in Nature 2020. Okay, so SARS-CoV-2 is much more closer to all the bat sequences. See, nowhere we are seeing the pangolin. Okay, I'm pretty sure there is no pangolin in between. I'm pretty sure bat has directly transmitted from either through fruits or through either through blood or because sometimes they even bite the animals. Okay, so okay, that must have been the major reason for transmission through bats directly instead of pangolin. So let me come out of the evolution part, and when you look at the contacts and contact tracing, because in India, okay, when you talk about the public health perspective, okay, India is definitely filled with a lot of cultural, traditional histories, and okay, we are having so many beliefs, superstitions, and this and that, and all the people are worried with a lot of social stigma. Okay, so they don't want to disclose any of their personal information, even they die, they don't want to disclose. They come out only at the 3rd stage, okay, primary contacts, okay, secondary contact, local transmission, and tertiary contact. And nobody is disclosing even till the tertiary contact in India, okay. Only when their immune system become worsened, and when they are down with fever, coughing, sneezing, okay, and only after that they come out and spell out that I am infected, okay. So until then, okay, he will experience all kind of symptoms, and he doesn't want to be hospitalized because of social stigma what Indians are having and carrying across them. They are carrying a big crown of social stigma on their heads. Okay, so I was talking about contact tracing and community monitoring is a very, very big problem in India. Okay, and they don't disclose. Even though they disclose, they will tell lies that I met only one person, I did not meet other 2 people. Okay, so other 2 people left without putting them in the quarantine or isolation, and that 2 people may infect other 2 people, and that 2 people infect 4, and 4 will infect 16 will infect 64, and then 256, then 1256. So this is how it goes on a logarithmic phase of infections. This is how the infection spreads among the people or among the communities. Okay, so I was talking about the R naught. Okay, so R naught is nothing but a reproductive number. It's basically calculated solely of how one person infects, one person of COVID infects 2.5 people. Okay, so this is called as R naught. Okay, this R naught has not mentioned about any of the government official in India. Okay, this has to be mentioned because when you talk about H1N1, a very big pandemic, okay, it is infecting only 1.6. It's infecting, Ebola is infecting only 2. SARS is infecting only 2 to 4. Okay, and now this is the old statistics. I'm pretty sure it must have even crossed 5 or 6, the reproductive number. Okay, this was an old slide. Okay, so one patient can infect so many people, and at the end of day, yeah, one person can infect at least 100 to 200 patients if he's an active carrier of the disease. Okay, so if you look at the global data of SARS-CoV-2 ongoing pandemic, look at the deaths occurring in USA, India, and Italy. Okay, so this is the kind of deaths occurring in Brazil and here in Mexico, South America, Spain, UK. Okay, so this has now even crossed so many deaths. Okay, these are the casualties. These are the cases which occurred. Okay, so in India we see 3.9. Okay, 3.9. It's a very big figure. Okay, so this is a USA data alone. If you see the New York, the deaths 31,159. This is the data taken from USA update on BBC, and it's very much updated information. Okay, next comes your New Jersey, and next comes your Illinois, and then Pennsylvania, and then Michigan, and so many other cases. Okay. So I was talking about genomic epidemiology. Okay, so there's so many countries. I'm pretty sure our Honorable Prime Minister Narendra Modi ji should spend some good amount of money for sequencing the whole genomes. I'm pretty sure in India at least 30 to 40 strains are really circulating among the 26 states, 28 states in India, and out of 28 states, okay, nearly 40 or 50 mutations must have occurred by now. So what we must do is we must really take the suggestions from the healthcare professionals, and what we have to do is we have to send at least 100 sequences every week, 100 potent cases every week to the central database, to the National Institute of Virology, and from there it should get sequenced, and from there it has to be placed to WHO or CDC, and this kind of database is maintained by next train team in UK. Okay, if you do this, we can come to know which is the major circulating strain across this country, and we can be aware of that, and we can also make sure that major circulating strain should not be mutated further. It has to be stopped, and that particular area should be quarantined, and that particular area should be under lockdown. Okay, so that is a major idea. So if we don't do that in India, okay, that we should experience much more cases. I'm pretty sure by the end of July, I'm pretty sure it will cross 5 crores. By the end of August, it will cross 7.5 crores only in India. Okay, if we don't do this, okay, I'm pretty sure we are going to end up in trouble. Okay, so in India, I think we have very less number of strains submitted so far. Okay, yeah, for the public database. Okay, so this kind of thing comes in 4 class. This is how it's moved from India as a blue line. Okay, this is Europe, Scandinavian countries, and Norway, Sweden, Iceland, Belarus, Ukraine. Okay, this is how it has moved from here to America, United States of America, and Mexico, and from there to Brazil, Bolivia, Paraguay, Argentina, Peru, Ecuador, Venezuela, and from here to China to Australia, Indonesia, Papua New Guinea, Philippines, Thailand, Sri Lanka, India. Okay, so we have received from China because we are very much more closer to China. Okay, so if you look into the number of strains in the genomes, their mutations. Okay, so what exactly happened is this is a polybasic site, cleavage site, which is directly linked to your all linked glycan residues. Okay, at the place of 681, 82, 83, 84. Okay, this PRRA, these are the more vulnerable sites for mutations in the spike protein. Okay, so there should be very precautions, especially this region should not get inserted, mutated, or recombined, and there should be enough care taken on these lines, especially. Okay, so if you look at this type, there are 2 kinds of strains, L and S type of circulating SARS-CoV-2 strains. Okay, The two major strains are nothing but, okay, L being the major type, it is circulating from China to Europe and America. They all belong to L type, and we belong in S type, is being the minor type. 30% of the strains circulating in India are, okay, we belong to S type. And especially this region, I'm talking about this region, L, S, L, 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 S, S, S. This region, this is the place where so many genomes have been sequenced.
Dr. Veerabrahmachari,
yes.
Can I just interrupt you for a second, Doctor?
Yeah, please. Yeah, please.
So we've just generated extension password and an ID. So your session just 2 minutes before 5, it will stop. We'll continue with the new password as long as your whole session will complete properly. So relax. You please, because it's a highly academic session and we'd like you to do justice. However, we will ask you questions and we'll take it properly, right? So 5 o'clock, you'll finish, and Dr. Veerabraham has sent you a new password and a ID. Is that okay?
Okay, fine.
Okay, thank you. Please continue. And 5 o'clock, you will stop, and we'll quickly log in. It'll just take a matter of 1 minute, and we'll get back. Right, sir?
So,
is that okay?
If you want me to stop here, and
no, because that session, I want to tell you before it ends. Ends at 5, and that continues from 5 on the new password. It'll take 1 minute to log in, right? I needed to tell you so that you're taking your own time. Thank you. Thank you.
Thank you. I hope my voice is audible to all of you. Yeah.
Maybe a little louder. I thought a little louder. I think your voice is very nice, but a little louder. Thank you.
Wait a second. I'll some water. Yeah. Shall I go on, madam? Shall I go on, madam?
Maybe 2 minutes. We're waiting for that 5 minutes.
Okay. So you want me to ask, want me to ask any questions in between?
Yes, maybe by the time, you know.
So we'll have a chat.
But very, very interesting. You've gone into such, in depth. And I'm not a science person. I'm a social scientist. But I listen to every word of what you are talking.
Okay.
Yeah. And
we are just in the ABCD level till now. So there is so much more.
No. I think you are XYZ level. I'm ABCD probably.
No. I'm talking about my slides, current slides. I'm still in my ABCD level.
Yes, please.
Okay.
You can go on fully because we'd like see when it's with all due respect to scientists and researchers, you should go your full length. That's when justice is done.
Because right? I always tell the people, because whenever you keep my lectures, okay, I should have good audience and I should have good ample of time.
Yes, true.
Because there is no point in, because the dissemination of knowledge will not be clear.
Yes.
I'm pretty sure that the goods will not be delivered aptly.
Yes, true.
That's the only thing. And most of the time, when many people call me, I'm very strict. I tell them I need very pin drop silence and very good audience.
Yeah.
And most of the time, I deliver lectures at colleges.
Yes.
The target audience are from arts department, and I go on speaking to the walls because nothing is getting to them.
Oh my God, that's even more distressing.
So no? I always ask the people, whomsoever ask me, I'm very strict, and I ask them, so how are the audience? Whether they belong to science, or do they really like science?
Yes.
So do they really want to listen? If they really don't want to talk, then listen. Okay, and I'm more than happy to come out of it. Because
so I think we just have 2 minutes. We have 2 minutes to cross your 5 days thing, and then log in with that.
Because I can't stop in between. There's a reason I'm just recreating and chit chatting in between.
I'm sorry.
Shall I go on, madam, or because you'll give me new password?
The new password, I think, is sent by Veerabraham, sir. Just check, sir.
So all of you check your chat box. We provide that feedback link and ID password. So all of you participants, please see that. Use that new ID and password. Please join after 5.
Please, all the participants, we apologize to Dr. Veerabrahmachari for this inconvenience. Technology, it happens. We are very sorry. And we ask our participants to come back because it's a very, very in-depth session. I don't think you'll hear another lecture like this.
Yeah, even I presume the same, madam.
I'm really, right? Please come back, and please, because the feedback session is not yet complete, and you must wait till he finishes. And that's the respect we have for academicians and researchers. We will all come back and listen to him finish and stand by for the national anthem, and then we shall go. So please, I think maybe it's time. You'll just 1 minute, and maybe you can just leave, and then just come back for the session.
So meanwhile, if anyone wants to ask me 2 questions for 2 minutes, I'm more than happy, because I don't want to go on and then stop and come back. Let me start again, start over from 2 minutes.
Do you have any questions?
Any questions to ask me? Till now, till evolution and till the structure, we haven't gone till structure yet.
We're presuming they don't have questions. They may have questions later. Thank you.
Okay. Okay, and next time when you, because today is Monday, and whole day I'm into several administrative positions.
Okay.
I'm tired at the end of the day, but still.
My God, I know. And hats off to your patience and your resilience.
Yeah, since morning I've been running
with so much of energy.
Yeah, exactly.
I appreciate that very much.
So it's only for the passion of science, passion for science.
Yes.
So
True.
I want the people to listen and at least learn